drug development and targeted therapy using biotechnology and using the knowledge that we know about the molecular biology. This is a sort of a practical part. I want to talk about the 2D gel electrophoresis, which is a sort of electrophoresis that separates the protein that I have in the tissue. It has two properties: separation according to the isoelectric point and separation according to the size. So we use it to separate a mixture of proteins. I have to get the normal tissue and the malignant tissue and compare both of them and see which protein and which DNA and which and what are the differences between both tissues. And I select this difference and I try to target a drug to destroy the protein or the DNA, which is the cause for this disease. Here it goes like that. We have the first step. During the first step, I separate according to the isoelectric point, and then I take the first strand and turn it over, and I separate it according to the size. I will have something like that. I will compare, as I told you, both the malignant and the normal tissue, and I will take what is different. What is different, I can determine it. What is the source of this protein and the DNA which has expressed this protein, either by polypeptide sequencing or by three-dimensional techniques or by mass spectrometry. When I know what is this protein, I have another technique which gives me, okay, I'm about to finish. Another technique is doing the DNA or protein microarray. I have a ship, and this ship has thousands of genes or thousands of proteins, and they are probes. All of it is probed with a fluorescent chemical. So I will realize that I found a certain protein which is highly expressed or a certain gene which is highly expressed in the malignant cell. I take this protein or this gene, and by my knowledge, I will think what are the compounds that can attack this active site of the protein, which is expressed by a certain gene. And I try all these compounds. I will find one of them will be promising. I have to follow up the ethics of doing, of developing a drug. And this ethics is to study it first on tissue culture to know the IC50. Then I study it on experimental animals to know the safety and the mode of administration of this drug. And then if it is successful, it will be promoted to clinical trials. Thank you so much. I tried to be in a hurry. I don't know you got it or not.
Thank you. Thank you, Professor, Your Excellency, Professor Zacharis.
Thank you.
Thank you very much. Back to the co-chair, please, Professor Zar.
May I invite Professor Dr. Adil Biltagi, Professor of Plant Pathology, former Minister of Agriculture, Egypt. His talk is entitled Navigation Navigation Towards the Future. That is yours, please.
Okay. Okay, thank you. Okay. Professor Dr. Mukhtar Zawahiri, the Vice President of the university, Your Excellency, His Excellency, Professor Dr. Amr Izzat Salama, a former Minister for Scientific Research, and the Chairman of the Arab University Union, Professor Abdelkader Abdelkarim, the Chairman of the University, Her Excellency Dr. Nadia Zakhari, I don't think it's, okay. Professor Nadia Zakhari, dear colleagues, the student community of the university, people who are virtually seeing us from other places in the world. When you have a voyage to the future and you navigate through this, you need to know from where you are coming, and sometimes you know where you are going. You have a dynamic targets, and these dynamic targets change according to what you would like to achieve, the problems which you have, and you want to really solve them. But since we were hunter and gatherer on this planet, and knowledge only kept human being driving through. When we talk about resilience, resilience does not come for people who are just laying down and they don't acquire knowledge. Resilience comes from acquiring knowledge. Then this hunter and gatherer has gone to agriculture and gone to industry, and they moved until our time through knowledge and knowledge only. Then we hear people talking about sustainability and knowledge-based economy and knowledge-based society. We need to say this is what was happening all the time. Without knowledge, we don't have any place to go to. Why this not is working? Okay, therefore we face a lot of challenges, and these challenges, one of the major challenges we face is the issue of global warming, which is affecting us all, and we will have all these repercussions because this planet, the carrying capacity of it is very limited. And this rupture happening because we will lose about 2 million hectares of land, loss of agrobiodiversity, increasing water scarcity, increased population and natural resources destruction by conflict and war. We will have the IPCC number 5 and number 6, which is the latest one, show up that there will be an increase in temperature. We were hoping after the Paris Agreement that we will go for 1.5 degrees. If you go through the report number 6, which was issued only last year, and a couple of other activities is going on, the answer is that we are going to go beyond 1.5 centigrade. Already, we are in 2 centigrade increase of temperature, more than industrial time, or industrial era. And therefore, areas in the dry areas will be affected. All this, the food, water, ecosystem, extreme weather event,
risk of irreversible change. All this is going to happen. The danger is that if we go beyond 2 centigrade toward the 4 centigrade, and this is what is the scenario now, you will have reduction in food production worldwide between 20% to 50%. Nowadays, we need, by year 2050, 70% more food, while you will have this reduction. And the world population is going upward all the time. We are heading from between 9 and 10 billion by year 2050. And at present, we are 7.9 billion. But the difference between the present and 2050, that 70% of this increase in population will be in poor countries, in 24 of the poorest countries in the world. And therefore, we are facing a big problem. Climate change will cause melting of ice caps, as well as it will threaten the deltas in the world. But these deltas were studied by the World Bank for 80 deltas. The most endangered delta are in the Mekong River, as well as the Nile Valley. Therefore, we will have a big impact in Egypt. And this is a scenario for 50 centimeter increase or 1 meter increase. And when you say 1 meter increase, this is the static water, not the wind and the waves. It could be, the 1 meter could go to 3 meters very easily by the wind. And therefore, the damage is measurable. Here, this man and his boy is 20 centimeters today. It will be more than 60 centimeters by 2050. Then the danger is real, as well as aquaculture will be affected, inland fishery, as well as open sea fishery will be affected. In the animal, for the animal issue, we will have infectious disease going between borders. It doesn't need any visa or a PCR or a certificate that they took vaccine. They can move and zoonotic disease is very dangerous. We know the bird flu and all that stuff, it will be easy transmitted. Therefore, this is a study by the Animal Health Center in Paris, but native plants as well is going to be affected. And we will have extreme heat, the precipitation will be affected, severe storm, more floods, more drought, as well as this impact will affect the fruits and its shape. And therefore, it will affect what we call post-harvest character. It will affect the disease spread in plant. And as well, it will affect the fauna and the flora. Even the biodiversity is threatened tremendously. And to do this, this is the classical gene bank in ICARDA, the International Center for Agricultural Research in Dry Area. I was the Director General of this place for more than 11 years. We cover all the dry areas worldwide. But meanwhile, this is another structure in an island called Svalbard. Svalbard is in the pole, and it has this structure which was built up by a platform called the global gene trust, and it will house about 6 million accessions, and it is in a glacial mountain whereby the seeds are kept inside this vault in the depths of about 80 meters inside, and it does not need any electricity or any other. It does not need any other issues. This is the vault from inside, and this is the board which was dealing with this. In addition to this, gene banks, which was spreading all over the world. This is in Baghdad. The one in Abu Ghraib was blown up. It was very close to the airport, and therefore all the genetic resources this place was going to will come back to the gene banks as the building blocks of new varieties in the future. Climate change will affect outmigration, and outmigration is expected to be between 200 million to 1 billion, and this, they call them the environmental refugees. This is a map for this, but we need, through the science and the gene banks, to have this NOAA boat to take us. Otherwise, the world is on the edge. And this is what will happen if we do nothing, if we don't use science and technology on this. What is the tools in front of us? It's all this list. But part of this list is biotechnology and genetic engineering and gene proteomics, gene mining, gene editing, or information technology. All these issues, including the nanotechnology and the biosensors and bioprocessing and the nanomaterial. And therefore, the nanotechnology will help on all these aspects. I don't want to get into details, but it's microarrays and nano particular complex of DNA and peptides, biological labeling and imaging, smart delivery system, pesticides and drugs, and agriculture and food system security and food quality and control. Let me go back and say, with this climate change, we have, as human beings, we have two things. Either we go for mitigation or we go for adaptation. To go for mitigation, this is the luxury of the OECD countries and the West. But for developing countries, it has to focus on adaptation. And adaptation has two major elements: a new genetical makeup, varieties which has traits of heat tolerance, as well as biotic and abiotic stresses, and the new agro-management techniques, on-farm irrigation, nutrition, and this could be wrapped up in what's called agriculture 4.0, which is smart agriculture or precision agriculture. And the massive program of human resource development, this university we are in now is part of it, which is education, as well as building up the human resources everywhere, not only for university graduates. Then it's a matter of knowledge all the way through, which we are looking for. In adaptation, for the new genetical makeup, there's genetic engineering, genome editing, developing C4 rice, nitrogen-fixing wheat, enhancing photosynthesis pathway, functional breeding, development of biotic and abiotic resistant varieties. New agro-management techniques, and we have a list of issues here which we need to do. The AI, artificial intelligence, as well as all the tools of ICTKM is going to be applied in the agro-management techniques. This is effort which is done by ICARDA for producing wheat which is tolerant to drought, Ageri here, the Genetic Engineering Institute at the Agricultural Research Center in Egypt, they had one line which has tolerance in wheat, as well as there was breakthrough in wheat that all the 21 wheat chromosomes was mapped, and this was a great breakthrough because it will increase the ability to rewrite the story of wheat crop improvement. Gene editing is very important. Gene editing could help us to really locate the sensitive genes and put the resistant genes in its place. Therefore, we can have more tolerant rice varieties by the gene editing. And I know that there is a special talk about this. Therefore, I'm not going to get into details, but this is a hope for the future for all of us. This is in Arabidopsis, whereby this transfer is happening, and then after that you take it from Arabidopsis to other plants. We need to go to this adaptation steps. We need to have this 4.0 farming, then we can optimize the food production as well as quantity and quality. Therefore, you have to go to smart agriculture, and meanwhile, you use a lot of tools like drones, proper water, optimizing water management, soil monitoring sensors, machine, etc. This is the Rna region. This is Central West Asia and North Africa. This whole area is very dry, and you see water poverty. All the countries in red, they have water poverty. Water poverty starts when you have less than 1,000 cubic meter of water per person per year. Therefore, any country which has above 1,000, it's okay. But when you go below that, Egypt now is coming to 500. Therefore, we are entering acute water poverty. Then the countries in the red here, they suffer from what we call acute water poverty. And of course, biotechnology has a lot to do with this, and the field, the advanced tools of it, to get varieties which could live in this place. And this is what we are hoping that it could be done in the future. We need to install modern irrigation. We need to go for this methods of really quantifying the change and detecting the change which is happening all the time in greenhouses or outside, but it's all the time you put sensors to look at that. Artificial intelligence will play a major role in this issue, and therefore, as we can see, that this issue is not static, because the increase in temperature will vary from one place to another. It's not going to be the same, and therefore we need to understand that we have to monitor all the time. This is hydroponics and so on. I don't want to get you in details of this, but this is part of the modern agriculture. And even in fisheries, there's a lot. This is algae production, which can be used here. What all the nations are seeking what? Are seeking to improve the coping capacity, because as you can see, all these countries here, they have hard time, as well as we need to look at the adaptive capacity and increase this capacity in different countries. We need to move from global to continental to regional to local, and this will help us to see what I'm going to put as plants in different parts, and therefore genetic engineering will help us to have this dynamic movement and move from one crop to another in different places and have this be scanned and cataloged, and therefore you can scale up and scale down, but always you can have an alternative for what you are planting in certain valley, and you can assess what will happen to this valley locally, because this is what will count. I would like to say here that a lot of input from science are really embedded there. The core is human resource development. You have to optimize the use of water as well as land. You have to look at policies and legislation, and you have to look at this in a very dynamic world. I don't want to get into details, but we have several agricultural strategies in Egypt, and we were, was done, but you need to fulfill this. Egypt has different agroecologies, and therefore it will be affected differently by climate change. All this will require the people who are working on introducing new genetical makeup to catalog this and try to cater for this issue. As an example, AGERI, which is Agricultural Genetic Engineering Institute, was established in 1992 here in Egypt, and 4 years before that it was a lab. But what I'm trying to say here, that we wanted very much to have the latest technology, like the next generation sequencing platform, to be introduced to the institute, and this has been done. And this will enable us to really look at what is there in our genetic resources and the genetic resources which will tolerate high temperature. And this sequencer, of course, at that time will really minimize the cost between $500 drop to 0.58 cent of a dollar. But it helped us to move forward. And I hope that in Egypt there will be more of these equipments to be used, because this effort is going to put a lot of demand in all of you in the field of biotechnology in the future. It is not even, you cannot really settle anywhere, you have to work hard. Let me go through this very quickly. What this means to the economy. The turnover in the biotechnology in EU 28, you have 2.3 trillion euro. And all this pharmaceutical, bioenergy, biofuel, forest-based, and as well as the food production, forestry, agriculture. Let us see very quickly. And this is the people who are involved in this 17 million person, and the value added, as you can see, $614 billion. This is the building of the bioeconomy edition, and just talk about Egypt. The information is lacking. It talks about drugs. It talks about the agriculture sector. It talks about the GMO. We have, since 1995, we formed the committee for biosafety. It's gone to the parliament and so on, but we need the law. We need the law to be implemented. Therefore, any GMO plants can go out and can be planted. We cannot leave the law lying, and a lot of effort has to be done to move forward on this. Steps has been taken, but this is how the countries in the world are. Lower middle income, Egypt is included, Egypt, India, Indonesia, and Vietnam, and the rest of the countries until you go to the high income OECD countries. What is the situation? This is a bioeconomy overall, 2.4 trillion, and here the people who are working into it. Of course, this will take you to a circular economy, whereby you optimize the waste, you bring it back again, all the time. I don't want to take more time. In the States, the bioeconomy there is 388 billion, 2% of the GDP. You will have crops is 104 billion, industry as well as the activity here, the distribution is in front of you. India is a very good example, and I intended to bring India because in India they are aiming at 150 billion bioeconomy by year 2025. Up till now, they arrived to 70 billion in the bioeconomy activities. And this is the chart for India. This is between vaccine, enzymes, biofuel, PT cotton, which is coming out of genetic engineering. Therefore, the price of doing nothing is like that. If you have a food crisis, you have political security and instability, you have financial, economical instability, and then you will disrupt the food security, and this is what will happen. All the resolutions we had worldwide, it failed to fulfill because lack of political will. We have Johannesburg, and 10 years after Johannesburg, and 20 years after Johannesburg, Kyoto, and then Cancun, Copenhagen, Glasgow, Paris. The translation, the box of translation does not exist. It's only we talk, we have commitment, it's not translated to action. And therefore, when I was in Copenhagen, they say this is how Copenhagen here. It's called Copenhagen, with no hope. And then we were in Cancun, they say, yes, we can. But there was nothing which was being fulfilled. Anyhow, this meetings has resulted in agreement that there will be a Green Climate Fund and technology mechanism. The first one is $100 billion. The technology mechanism to allow scientists like we have here, scientists from everywhere, scientists from the whole world to work with each other in problem of modeling, assessment, and all aspects which relate to this. Adaptation it looks like the game of snakes and ladder. Therefore, without knowledge, you could go and head for the head of the snake all the time, rather than going to, what is this? Put this last slide. Okay. Therefore, this is what we are hoping for. We hope that international and national organization will link, as well as Professor Ahmed Salama work with Arab universities. I hope that they link like that, as well as other networks in the world, because this is the only hope we had. We have to glide through all this circle, and this circle here is managing implementation process, and it has to go through information, learning, incentives, and all these aspects has to come together. Therefore, we can move forward. They're working together. Each element of this has to have its own indicator. It has to be measured, and we have to see the way forward. Therefore, people like this, and Nobel Prize, the only Nobel Prize winner in agriculture is Professor Bolog, standing there at ICARDA, but these people has goodwill for change. What we are aiming at is peace and stability and prosperity. Therefore, the distance between the have and have not has to be bridged, but through knowledge and resilience and through efforts which needed to be done, regardless what is the race or class or gender or age or ethnicity or disability. This is what we should head for, because this differential issue which is happening all over the world is really a sad story. We need to have a shift of consciousness on the part of the have and the have not, and a new understanding of the oneness of humanity. I do thank you. شكرا يا فندم. طبعا انا اسف. لا شكرا تسلم.
Just a minute, Dr. Mukhtar will have the headset.
Let me present Professor Dr. Mukhtar al-Zawahiri. He's a professor of molecular medicine, College of Biotechnology in Must University. And he worked on the title of his presentation will be Stem Cells and Regenerative Medicine. We are all looking forward to hear this presentation, and stem cells give a lot of hope for issues which Her Excellency Professor Nadia Zakhari even talked about. Professor Zawahiri, the floor is yours.
Thank you. As we know, aging is a slow process during which the body undergoes changes that eventually bring about death. ولكن even if there is no marked disease is present. So scientists are trying to extend the human lifespan and the health span, which is the length of the body functions, the length of time the body functions and functions normally. Okay, transplantation from human donors restricted by the scarcity of donor tissues, so stem cells could provide a source of alternative graft materials. Then, what are stem cells? Stem cells, like other cells, they proliferate, they remain exquisite, apoptose, senescence, adhere, and migrate. And they have two more properties, which are the capability of self-renewal or renew or proliferate in an immature state. Second, the ability to differentiate into any cell type. In addition, in many tissues, they serve as a source of internal repair system. And this is the difference between the adult somatic cell and the stem cell. In the adult somatic stem cell, in the adult somatic cell, the cell divides into two identical daughter cells. But in the stem cell, the cell has the potential either to remain as a stem cell or to differentiate to a specialized cell type. What are the types of stem cells? We have three types of stem cells. The embryonic stem cells, which are pluripotent stem cells, the adult stem cells, which are multipotent stem cells, and the induced stem cells, which are pluripotent stem cells. They are similar to the embryonic stem cells, but we can do it in vitro. We can induce it using the reprogramming system. About the embryonic stem cells or pluripotent stem cells, we have to understand the process of embryonic development, which are the fertilization, the cleavage, differentiation, morphogenesis, and growth. To understand the commitment or the potency commitment of the stem cell during the embryonic development, to acquire the different techniques in vitro to use it in the clinical application. Fertilization begins with the one sperm fertilizes one ovum, resulting in a fertilized egg with two pronuclei, the male pronuclei and the female pronuclei. In this stage, in this stage, it is called pre-embryo. So after fusion of the two pronuclei, we get the zygote with the diploid nucleus containing the 46 chromosomes, human chromosomes. And at this stage, this is the only totipotent stem cell. This is the embryo, the beginning of the embryo. And this is the only cell which has the totipotent stem cell, which means it can produce or it can form all the tissues of the embryo and the extra embryonic tissues. Then cleavage occurs in the zygote in the pre-implantation development. The zygote divides into two cells, then four cell embryo to four cell embryo, 8 cell embryo, and to the morula. Then we get the blastocyst. The blastocyst, this is the blastocyst after morphogenesis occurs, and the blastocyst has the inner cell mass أو اللي هي or the embryonic pluripotent stem cells, which are about 100 to 150 cells, and the trophoblast, as you can see from the figure. Then differentiation occurs by the expressed versus unexpressed genes. Morphogenesis occurs during the embryogenesis. After the result of the cell division is a block of cells, but the morphogenesis, in the morphogenesis, the apoptose gets rid of unwanted cells to induce the organized embryo as we can see. Then growth occurs, and we get the whole body of the human by its systems, its organs, and so on. So life was much simple when I was a stem cell. There is the potency commitment of the stem cell during embryogenesis, which are the zygote. During the zygote, this is the totipotent stem cell, and the embryonic stem cells after the division, after the proliferation of the zygote, we get the inner cell mass, the embryonic inner cell mass in the blastocyst, which are embryonic pluripotent stem cells. Then, after differentiation, we get the adult stem cells or the somatic stem cells. In the somatic stem cells, we can find some of them are multipotent stem cells, and others oligopotent stem cells, and even unipotent stem cell. And we have here in the bone marrow cells, this is adult somatic stem cell. The stem cell here is a multi-potent stem cell because it differentiates into different types of stem cells, progenitor cells, during the blood formation أو الهوموبيوزيس. وبالتالي هنبص هنا and this are the oligopotent stem cell, this one, the oligopotent stem cell, because it differentiated into 3 types of blood cells. This is a unipotent stem cell because it differentiates into 1 type of stem cell. The sources of embryonic stem cells. For the embryonic stem cells, we have 2 main sources: the in vitro fertilization embryos, or blastocyst, indeed, and the therapeutic cloning blastocyst. In the case of in vitro fertilization, of course, it contains of stimulation and the retrieval of the eggs, then the sperm collection, in vitro fertilization, and the implantation development of the embryo. We get the blastocyst. During the therapeutic cloning, we take the adult cell from the patient and the unnucleated, unfertilized egg from the donor, and we do nuclear transfer, our nucleus transfer, and to get the zygote. In both cases, we use the in vitro pre-implantation development. As you see, the after fertilization, zygote gives the 2-cell stage and so on until we get the blastocyst. This is the blastocyst. This is the stage at which we get the embryonic stem cells. Adult stem cells, for the adult stem cells, which are multipotent stem cells, we get them from different, these are some sources of the adult stem cells. We get it from the bone marrow cells, stem cells, cord blood stem cells, fat stem cells, mesenchymal stem cells, epithelial hair follicle stem cells, and amniotic fluid stem cells. In that stem cell research and the strategy approach, we have to understand the factors which involved during the commitment of the stem cell to be proliferated and differentiated into the different types of body cells. A family of these growth factors are controlling the cell cycle progression. As you see, we have two big families of cyclins and cyclin-dependent kinases and cyclin-dependent kinase inhibitors. We have the karyokinesis, including the synthesis of DNA. So the group or the families of these growth factors are called S cyclin, S cyclin-dependent kinase, S cyclin-dependent kinase inhibitor, and so on. And during the cytokinesis, or including mitosis or meiosis, they called M cyclins, M kinase, and M kinase inhibitors, and so on. We also have a group of growth factors controlling the progression of cell, and at the same time, they check if the DNA is correct or there's any damage in the DNA. So we have this series of growth factors. We have also growth factors controlling the gene expression to produce the required proteins during the expression versus unexpression genes to get the different types of cells. So we have the stimulatory pathway, and the inhibitory pathway. The inhibitory pathway includes growth factors which are called proto-oncogenes, and the inhibitory group of growth factors are called tumor suppressor genes. And both of them has three main groups: extracellular growth factors, then receptor growth factors, then transducer growth factors during the cytoplasm, and the pathways which reach the nucleus, and inside the nucleus there are the nuclear growth factors, which are the transcription factors. So we have very large number of growth factors responsible for the proliferation and differentiation of the stem cells to get the specialized cells. So we have to understand the role of such growth factors. This is a growth factor in the top, and their growth factor receptors, and these are the pathways during which the transducers are functioned, and then the transcription factors which switch on or switch off the gene to be expressed for a specific function to get the differentiated cell type. So we have to understand that the stem cell technology, or the stem cell research technology, is dependent on the programming, genetic programming, and the genetic reprogramming. Genetic programming of the embryonic stem cells and reprogramming of adult somatic stem cells, at which we can use it to induce the pluripotent stem cells. After that, this is a technique which we use to get different types of specialized cells from the stem cell. We take the blastola or blastocyst, and in which we can see the inner cell mass or the embryonic stem cells. We take the inner cell mass and then dissociate them to get the embryonic stem cells. And we can culture them, and we can keep the culture dish in the incubator. After that, we can do subcultures وبعدين ممكن نعمل cryogenic to serve them. Then, if we want to do the differentiation to any type of wanted cells, we add different growth factors according to the functions of the growth factors which we have seen before. So we add the growth factors required for differentiation of the stem cell to any cell type, such as muscle cell, blood cell, nerve cell, and so on. And this is the difference between the pluripotent stem cell and the multipotent stem cell during the cell proliferation and differentiation. For the induced pluripotent stem cell, because it's very important, was to use the embryonic stem cells taken from the in vitro fertilization or from the therapeutic cloning. It is somehow difficult, so we can induce the pluripotent stem cells which are similar to the embryonic stem cells in vitro, using the reprogramming of the adult cells to become a stem cell again. The induced by imprinting reprogramming of the adult cells. These are the group genes that are forced, the nano, CMYC, أو SOX2, LIN28, KLF4, OCR. These are يعني هم المين group of cells used in the induction of pluripotent stem cells. We take the somatic adult cell and use these genes for the reprogramming and get the induced pluripotent stem cells, as we can see. And in the regenerative medicine, there is a potential use of stem cells in research and in treatment of the disease. This is some health problems that might be treated with or using the stem cells. The neurological diseases, limb amputation, heart disease, pancreas, أو diabetes mellitus, spinal cord injuries, burns, and genetic diseases such as cystic fibrosis, Huntington's disease, and other diseases. This is an example how to treat a disease. If we have a patient, we can use the adult cell from this patient using the technique of therapeutic cloning. Then we get a nucleated ovum from a donor. After fertilization, we get the zygote. The proliferation during the pre-implantation development, in vitro, we can get the embryonic stem cells from the blastocyst, and then we can use the required growth factors to get any cell type after their differentiation. And we have to use this cell type according to the disease of the patient. We can treat the patient. And look, here it is very important, this technique, to be used because it is a self-cell. Yani self-stem cell. I got self-stem cell using the induced pluripotent polypotent stem cell because it is immunogenetically compatible for the patient. يعني transplantation هنا not rejected. So it is very benefit to use such a technique. This is a model, a mouse model of sickle cell anemia disease. And as we see in the steps of that technique, we take the adult cell from the skin cell or any other adult stem cell from the mouse. Then we use the growth factors for the reprogramming to get the induced pluripotent stem cell. And then we do gene therapy to correct the mutation of the patient because it is present in every cell of the body. And when we take the adult cell from the patient, it contains as well the gene of sickle cell anemia. So we have to treat it. And we have to treat it in culture to correct the mutation, and then proliferation in the tissue culture. And we take the normal cells after the correction of the mutation to the mouse, which recovered from the sickle cell anemia. Thank you.
Thank you, Professor Zawahri. And now we'll go for our last speaker in this session. And this is Professor Nagla Abdallah, Professor of Genetic Engineering, College of Agriculture, Cairo University. The title of her presentation is Genome Editing Technique in Plants. Professor Abdallah, the floor is yours.
Just need to. ممكن تفتحها لي؟ مفتوحة؟ اوكي شكرا. Thank you. طب اوكي كده. Okay. Good morning, everybody. Thank you for the invitation to participate in this successful conference, and I wish you all the best. Thank you for the president of the university and the president of the conference, and for all the president people. Today, I'm going to talk about very important topics. It's about genome editing and how can we use it to fight hunger. So, not sure, pointer is not working. Okay, my topic is genome editing techniques in plants, further perspective toward zero hunger. As we know that MENA region It comprises low and mid income, and the population is very high. We have about 296 million people, 120 million in rural area. We have about 28% depending on agriculture, and the area is characterized by an arid and semi-arid land. That's why we need to find a way to end the hunger and malnutrition. Today we have more than 800 million people around the world go to bed hungry each night. And the global change, of course, and the COVID-19 is one of the factors that affect and hinder the world Sustainable Development Goals, which was expected to end the hunger by 2030. How will we produce enough food for the expected 2 billion additional people who will join in 2050? We can do that. We can do that as there's climate change are getting worse every year. We have a lot of problems like biotic and biotic stress, and threat for all of this threat for our food crops. So the main objectives for natural agricultural resources is to have a zero hunger, and this can reflect 5 elements for the Sustainable Development Goals, which is sustainability for natural resources, and improvement of agriculture productivity, expanding of the use of modern and well-developed irrigation techniques, promoting land reclamation, and sustainable of development of rainfall feed agriculture. So goal of crops improvements, we can improve crop improvement could be help in eradicating hunger, eliminating of poverty, sustainable management, and utilizing of natural resources. So new plant breeding techniques, as we all know, started with the genetic modified crops, and we have after that we genome editing. It arises as models for trying to solve a lot of problems that we face, especially in agriculture fields. So biotechnology products have highly profitable for farmers and society. Commercializing of GM crops started in 1996, and until now there is a lot of countries are practicing and commercializing this product. Make sustainable advances in our knowledge of the diversity of some crops, application of new breeding techniques with the additional techniques lead us to more impact genetic resources and biodiversity. So as we know, new technique of genetic modified or genetic engineering product is not affecting biodiversity. No, it will help biodiversity to keep the different variety that we have. Sorry. So modern biotechnology is a new aspect of agriculture and has many applications, and this required studying the following: impact of the application of biotechnology in crop and livestock production, socio-economic impact of biotechnology on world trade and economic development, managing intellectual property, risk assessment to release of modern organization into environment, cooperation of private and public sectors. We cannot do that without the approval of the private, of the stakeholders and decision maker to allow us to use this technology. So biotechnology, as we see, started with crossing between different species or different plants to transfer one characters from one plant to another. It may take 10 years to transfer this character and stabilize the other wanted characters. We can use mutation, like they developed citrus, seedless citrus. We can use monoclonal variation. And the most important is transgenic and gene silencing and genome editing. So crop development, as we know, transgenic plant, it may take about more than 10 years to get the plant commercialized. We have to develop this plant, it may take 3 to 5 years to have a homozygote plant that have the desired character, and then to get approval from different committees until we get the final commercialization. So this technique is very successful, but unfortunately it's take a lot of time to get benefit from this product, and it costs a lot of money and a lot of effort and time consuming. In the beginning, this was the file was required to get approval for the regulations from the regulating committee. Right now, we only need one dossier to submit it for the committee to get approval. So at the beginning, there was a lot of people anti-GM that work against the new technology and hinder the development of this technology. That's why genome editing started to arise. Genome editing is an easier, faster, does not have any foreign DNA. It can be replaced in a lot of things in the genetic modified, but until now it's not 100% can be replaced. But at least we can avoid a lot of obstacles that we meet with genetic modified crops. So genome editing, as we call it, site-directed nuclease. It's site-directed nuclease because it's a protein that can cut the double-stranded DNA and modify it. So we have different kinds of site-directed nuclease. This is the four common one: the zinc finger nuclease, the TALENT, the meganuclease, and the CRISPR. The first three, zinc finger with TALENT and meganuclease, depend on the protein to target the site we want in the genome. So we have the whole genome with a million of nucleotides. We target a specific site in the genome to make an editing on these parts. So it was very complicated to use the meganuclease and the zinc finger. TALENT is less complex, but the CRISPR is easier because it depends on RNA to target our sequence. It's very easy, very cheap, and any small scientist can do this work, and less expensive, of course. So most of the work, we can find the publication on most of the work on the CRISPR for modifying this strategy. So meganuclease started in 2003, TALENT started in 2009, CRISPR in 2013, but the first product plant was developed in 2012 by the TALENT in rice. So we have a lot of techniques developed each year. We have a new technique, as we're going to see, that it's not only the CRISPR-Cas9. CRISPR-Cas9 depend on a Cas enzyme that cut the DNA with a short, very short sequence of RNA. And we have the CPF1, the edit nuclease, the single-stranded target editing, and we have the epigenetics and the prime editing. For the CRISPR-Cas9, as we said, it needs the Cas enzyme. We can use the wild-type Cas enzyme. We can use the nickase. Instead of cutting the two strands, it can cut only one strand. And you can see we have a lot of application came from the nickase. And we have the dead Cas9. All of this can use it for editing. We have the Cas12 or CPF1. This is more efficient than the Cas1, Cas9. Cas9 has a little drawback because it has something we call it off-target effect. Off-target effect, if the target RNA is similar to another sequence of the genome, it can edit not only the target genome, but also in another site. For the Cas12 is more efficient, does not need the sequence, we call it the PAM, which is NGG. It needs another sequence, we call it TTN, and it cuts the DNA into a sticky end, so it's more efficient than Cas9. Both of them are used for genome editing, and you can find a lot of publication come from this work. Base editing. Base editing is another revolution for the genome editing because for the Cas9, it cut the double-stranded DNA, and it does not religate by itself. The DNA cannot be broken, so it has to be religated. To form religation, it should insert or delete few nucleotides. So insertion or deletion will cause frame shift, and the gene will be no longer active. So we'll have a mutation. So if you want to suppress this gene, we can use the genome editing Cas9. For the base editing, it's very precise because you can choose the one nucleotide you want to change it and change it with another nucleotide. So it's not random like the Cas9, because if you want to change one amino acid or make a stop codon, you can choose one nucleotide they want to change it, and we will change this by the base editing. And base editing is very new also technique and has a lot of publication came from it. Okay, we have also this for the base editing, an example for which came from this for RNA. Sorry. So not only work for DNA, but it also work for RNA. So there is another genome editing. We can use an RNA editing. That means that we can edit the genome of the virus, like the COVID-19. If you want to make a stop codon in the genome of the virus, we can use the genome editing to stop the function of the gene or make any modification in this. So this is an example. We use the Cas13. The Cas13 is specific for RNA. So they use a target with the RNA that can identify the RNA that we want to edit. Prime editing. Prime editing is another thing because if you want to make a substitution, we want to change part of the gene with another part. So we have to use a template or oligonucleotide to make a switch and substitute this part. For the prime editing, no, we can use the RNA with the sequence of the part we want to use it to substitute in the genome. And we use it with it reverse transcriptase. So in that case, the RNA will transfer to DNA and can switch the part that we want to use. So the prime editing is another new revolution for the genome editing. So as we see each year, we have a new technique coming out from the genome editing. Epigenome. Epigenome, that in that case, they use a dead enzyme. So the enzyme will not break the DNA, but it will target the protein into that specific site in the genome. In that case, as Dr. Nadia said, we can use it for methylation or demethylation. If we have a gene inactive because of the methylation, we can put an enzyme to demethylate the gene and turn it into active form. Or if we want to inactivate a gene, we can also use genome editing. So epigenome, we can use it for methylation, not only the DNA, also the chromatin. And it is inherited. So once it is introduced in the genome, it will be inherited in the next generation. So this is an example of what genome editing can be performed until now. What about organelles? Yes, we can use genome editing for organelle editing, but in that case, we cannot use Cas9 because Cas9 need the protein and the RNA. If we express the protein in the nucleus, we have to join the protein and the RNA and force them to enter into the organelle. In that case, they use the TALEN because the TALEN use the protein that can be target the DNA, and we use site-directed the polypeptide to target inside the mitochondria or the chloroplast. In that case, we can modify the genome of the mitochondria or the chloroplast. So there is a lot of application of the genome editing, and each year we'll hear a new technology that can be applied for genome editing. So we just give you a brief about what can genome editing do. What about the application? There is a lot of application. Since the first product came in 2012, we have more than 500 papers published about genome editing in plants. It's also applied into animal and for human. It also have been used for curing some, for clinical trial, for leukemia and for cancer, like and HIV. There is a lot of publication came from that. It need approval from the patient itself to approve using this technique. So for CRISPR-Cas9, we'll concentrate on CRISPR-Cas9 because it's most of the publication came from CRISPR. So it has been applied for virus resistance, disease resistance, physical and chemical resistance, crop protection, enhancement, manipulation of bioactive compound product, and all of that has been applied by the CRISPR. These are some examples that's already published in the literature. We have oil quality reduced, saturated fat. We have bacterial light resistant aroma. We have reduced acrylamide chloroplast, visible gene market like in corn. We have powdery mildew resistance. We have also waxy corn. This corn has been used by silencing the gene expression of wax. We have non-stem tomato, so the tomato can be formed without stem. It have a lot of products, and a lot of companies preferred using them. We have high oleic acid soya. This have been used by deletion in Fed 2. Sticky rice. This is in the Far East. People like to have the rice sticky, and this product is already in the market. The gamma amino acid in tomato. Gamma amino acid in tomato has been already published and commercialized. So it's been commercialized in Japan, and they already have a company producing this product. And they said that this product have a good benefit from using it, like reduce blood pressure and reduce relaxation and factors as a neurotransmitter and a lot of things. The only thing that reduce the concentration of the GAPA is that it has a polypeptide at the 3' end that degrade the protein very quickly. So they use the CRISPR-Cas9 to stop the expression of the 3' polypeptide, and in that case we have a high product of the gamma in the plant. It was very easy technique, and it's now already in the market. So here we have some examples. This is in the already in the book is going to be arised as under publication. So we have a lot of products started from 2012 with the first rice light resistant using the talent, and we have a lot of application since then in different plants. So I'm presenting here the first plant produced by genome editing. Here we also have, this is not my application, this is a paper published in 2020. They show us that there is the different crops they can use genome editing for modifying starch, for modifying protein content, for modifying lipid composition, for increasing functional metabolite, for decreasing anti-nutrition and enhancing market value. So if you start talking about the application of the CRISPR, it will be endless. We have here in our paper we published this year about genome editing techniques in plant, a comprehensive review and future perspective towards zero hunger. It has like a database, small database about the plant already published or commercialized it using genome editing, and it covers all the techniques if you want to go through it. It's in GM crops and food, and I'm editor-in-chief of this journal. What about animal? For animal, also they have been used for genome editing in animal, like in cow, they produce hornless cow. So they mutated one gene and the cow will no longer have horn. Sorry. Also for parasite resistance, they also use it for parasite resistance, like screwworm larva, resistant for that. Also for climate change in cow, they use it to allow the cow to stand a lot of temperature. And for fish, of course, to increase the meat and the size of the fish. This is a lot of application, and a lot of product came from fish, and we have already fish in the market for commercializing with high expression, with high size and high meat in them. Here also another example in animals and different application. They have been used in pigs, in cow, in sheep, in monkey and dogs and so on. And this is a different technique. Even the base editing has been used for that, for substituting one amino acid with another, with the Cas9, with the base editing for RNA. What about in Africa? So Africa already using this technique, and they have a lot of application of using the genome editing. And there is an ISA brochure showing the application of the genome editing in Africa. In Egypt, we have 2 projects, as far as I know, one in Cairo University and one in Agiri. In Cairo University, we use genome editing to increase drought tolerance using the CRISPR-Cas9. And in Agiri, they use for sorghum to to reduce the lignin content. But here we have another example in different country in Africa. What about this challenge? Of course, anti-scientist people came and tried to stop these techniques. But if we see there is some techniques that there is some applied product that cannot be detected, like epigenome, or if you use base editing, you change one nucleotide with another. How can you identify that this is already edited plant or modified plant? So, of course, the scientists should have an ethical issue and say, I have developed this plant using the genome editing. And to commercialize it, we have to show how they already produce this technology. So the main obstacle that we face is the regulatory pathway. The absence of clear guidance. There is no guidance to say, if you do that, you will have this right or not, to be applicable or not. It depends on different country. Each country have its role and its pathway to allow this product to be commercialized and to be transboundary transferred. So if we have an editing plant in one country and we want to transfer it to another country, there's no regulation that prevent or avoid this technique. The weakness in assessment, inspect, and enforcement capacity, limited capacity for identifying present products, propose border, of course, as I said, across the okay. For genetic regulations, we have conventional method. Of course, this is not regulated. If we use different breeding, if we have genome editing, we have 3 types. So the EFSA and the regulatory system, they divide it into 3 types. Side-directed nuclease 1, 2, and 3. So 1, if we use a substitution, insertion, or deletion for few, substitution, insertion, or deletion of nucleotide without using any foreign DNA. In that case, we call it SDN1. SDN2, if we use very small templates for editing, in that case, the change is very limited. Type 3, if we introduce a gene or part of the gene that's not presented in this plant, in this case, we call it side-direct type SDN3. And we have the GMOs, of course, this should be regulated. So genome editing, we divide it into SDN1, 2, and 3. Most of the product is developed by SDN1 because it's easier. Here is the paper published showing the different country that allow the regulation methodology in the genome editing. So this country shows, the green one shows that they allow commercialization without regulation for at least SDN1. Some countries allow SDN2 also. The yellow one is discussing ongoing, like in England and different countries in Africa and in India and so on. And in the red, they consider any product came from genome editing as genetically modified crops. So here we have an example for genome editing in Japan, which already have product in the market. They consider SDN1 and 2 not regulated. 3 it should be regulated. In US aid, of course, they have a lot of another problem that they have 3 organizing committee that regulate any GM crop like the SCA, FDA, and the EPA. So President Obama in 2018 make like a network between the 3 organizing committee and make a decision that they all have its own specification to allow the product produced in the markets. And by the way, he changes the name from genetic modified or genetic engineering to bioengineered crops. So bioengineered crops, so if you go to the market and try to find if this product is already transgenic or not, you don't look for GM crops, you look for bioengineered. And here we have, we have some important products that have been applied using the genome editing. For commercialization, a lot of products have been examined or approved for commercialization, but not all of them are commercialized. The number of commercialized plants is very limited, and the first commercialized plant was in 2019 with high oleic soybean. This was in the United States, and we have another example, the GAPA in Japan. We have the waxy rice also in the Far East, and in soybean. We have a lot of products already commercialized. And in China last week, approved genome editing, crop engineering research for fungal resistance. I think it's fungal resistance. Anyway, this product will be expected to be in the market very soon. As conclusion, we can say GM crops are a topic of much deliberation and attention, very prevalent in some part of the world and abandoned in another. GM crops, genetic engineered or genome engineering crops are required for meeting food security in today's growing population. Public PPB technology are required to achieve zero hunger. And the genome editing hold great promises and set of to transform health and agriculture sectors. Of course, as we said, genome editing is not a magic stick. It's still in the beginning. It only have 10 years applications. So we expect in the next 10 years, we'll have a lot of promises coming out from this work. And thank you very much for your attention.
Thanks, Professor Nagla Abdallah, for this good presentation. And I hope that this legislation issue will be done and then we move forward. I would like to ask the organizer if there is any special announcement. Thank you.
Thank you, Doctor. Thank you for you all, dear colleagues. We are already beyond the scheduled time. So I will beg your pardon just to minimize the time for coffee break. And instead of 15 minutes, it will be like 5, 7, something like that. So I hope Perta will manage the time. We will start, inshallah, the second session on time. So I hope you just collaborate with us to minimize the time of break, and then, inshallah, we will proceed on time. Thank you. مرة تانية برحب بحضراتكم. من فضلكم be seated. All guests attendees, please be seated. I will beg your pardon again. We need to compress this session just to go on the schedule. Please be seated. اديني confirmation دكتورة وجيدة. I would like to ask Dr. Wael Abul Khair and Dr. Wagida Anwar and Dr. Perta and other guys in this session just to compress their lectures for 2 minutes. And these 2 minutes will make a difference in the schedule, inshallah. I beg your pardon, Dr. Wael, please. And actually, I asked Perta the same, just to compress the time for 2 minutes only. Just 2 minutes will make some difference at the end of the day. This session, session number 1, is chaired by Professor Wagida Anwar and Dr. Perta from Copenhagen University in Finland, and then the chair with Professor Wagida. Professor Wagida, please.
صباح الخير أو مساء الخير. يسعدنا إن إحنا نبدأ الجلسة الثانية وهي التشير بيرسون إحنا والدكتور بيرتا هوتلنان. I'm very happy to introduce her to speak as a first speaker. She is assistant professor of biomedical science, University of Helsinki, Finland, and she will speak about dendritic spines, the memory units of the brain.
Okay, can you hear me now? Yes. So thanks a lot for inviting me here. It has been really nice experience already so far. I normally start with this slide, which introduces our lab work, and we try to use the biotechnology in principle to understand the brain. And I have a background in cell biology, so we use a lot of cells. And for example, we mostly use the primary rat neurons, what we dissociate every week from the rat embryos. And we can manipulate them with many biotechnological means, what you certainly know. I like a lot by myself with live cell imaging. We had here a video, but it has run till the end. And we also do the behavior and also imaging. We can image the brain of the mouse. So we can come from the molecular basics, from biochemistry, even from the cell tube, protein chemistry to the cell animal behavior. I wanted to start with the groups because this is also binding why I'm here and what is my relationship with Egypt. Here today, I will mostly speak about the work of Pushpa Khanal, who is one of the Nepalese PhD students, really excellent. But I wanted to highlight from the past members, the Amr Abu Eles, who is here in the middle, and he was the very excellent Egyptian student who just graduated 2020 from my group, and we had a very nice 9 years actually together. He started as a master thesis student. Maybe a kind of a power of the writing nice letters. Amr was just writing me a letter and asking if he could join my group. And this letter was so good, and I decided immediately, yes, you can join the group and let's start work together. And it was really a joy. About Amr, one more thing, he started studying in our group the axon inertia segment, which was the new field for the whole group, and he set up the super resolution methods. He did actually many things for that, and it was really great. So let's continue with what I will speak today. So also during this conference, I hope you will learn many things and your brain will change. So the question is, what is changing? We often maybe don't think so details, but because I'm cell biologist, I think about details. This is an easy version of how brain works. Brain always predicts, it collects the data. As I'm collecting now, I see all of you, but I don't see maybe you so clearly from here, and my brain kind of predicts who you are and who you are maybe not. But if I would like to train my brain and change so that I would see better who is who, I should kind of give the brain the right answers. And this is the picture telling here. So these are the neurons who collect the data. It can be sometimes very blurry, so it's not clear. Then there are kind of neurons in between who bring the signal forward, and then there is a kind of output. And if everything is correct, fine. But if the brain finds that this is not yet totally correct, it needs to change something. It needs to train these connections between neurons, and then the output will be corrected. I explain this one more time with those who have glasses. This year I came so old that I had to buy reading glasses. And if you see the text which is too small for you, it's blurry, and you have hard time to know what is written there. But if you then put on your glasses and you see clearly what is written there, and then you take out glasses again, suddenly your eyes will see what is actually written there. So your brain kind of know better how to predict this blurry thing, what you see with your eyes, and then you have trained your brain to see properly. So then coming, what is changing and how to change these connections, which are very important for the learning of the brain. And here we come to my research. So let's start from the brain slice from the mouse here. And then we zoom in. This is, for example, the area called hippocampus, which is very important for the learning. And if we zoom in to the neurons inside the brain, we have colored specific neurons with the GFP fluorescent protein, and then we can look at them with the fluorescence microscopes. And in these cells, you can see these very small protrusions here, and they are called dendritic spines. And they are actually those memory units which you can change, and by changing them, you can change what your memory is storing or your brain is storing and how you have learned things. This is another type of cell, and it's very nice dendritic spines. Let's go forward. These are now the hippocampal neurons, what we normally work on the cultures, and they express the GFP actin, so we can visualize them nicely. And we can zoom in, and we can see this nice dendritic spines closer. And this is the typical shape. They look like mushrooms. They have a thin neck and then a bulbous head. And why they are important is that, first of all, by changing the number of the spines, we can change the connections between neurons, and also we can change by changing the size of these spines, we can change how strong these synapses are and how much weight they can put for the, for example, skills what you have already learned. And most importantly, there are synapses in these spines, and the spine size and number affect the function of the neuron. I tell a few more examples of the learning. Research, this is the first experiment showing clearly that when we learn something new, there will be new spines appearing. So this is very simplified version of the connecting spines, the learning. Always when you learn something new, you need to add some new spines and new connections between neurons. And this is the experiment where researchers are first imaging the same dendrites through the skull or through the glass in the skull in the mouse, and then the mouse will be learn some new task. And if they image again the same dendrites, they can see that there are new dendritic spines appearing. There are also other experiments where they could show that when birds are learning to sing, they all have their own song, and they first, when they learn, they mimic the oldest bird. And researchers could follow how the dendritic spines, they were changing their places, and there was a high turnover rate of spines during the learning. But when the song was good and the bird has learned the task, the spines were stabilized and then they stayed. So it's not only adding or deleting spines, but you can also increase the spine size. And what is important in increasing the spine size is that you add the receptors for the synapses, which makes the synapse much stronger, and then it has more value in the whole competition. Or it can also make the spine smaller and the receptors less, and then you lose this connection. So there are many ways to compute in the brain. This is the simplified version. So you have an original situation, then you have a learning, you have new spines. And when you repeat the learning, you really train this new skill. You can grow these spines and synapses, and then they will be more stable, and you maintain your new skill. And at the same time, when we add new spines, normally brain will delete some spines and synapses which are not important. So this is during learning and repeated training, there will be other spines which will disappear. If you have too many spines, this will bring you to the problems. And for example, in autism spectrum disorder, the people have too many spines, so you can understand that too many spines is also bad. It needs to be well regulated. So we thought in my group that neuronal activation and formation of new connections are crucial for brain functions. This is known for many years. And although we know that these both are very important and they are somehow linked, they need to be linked, we didn't know any molecular mechanisms or cellular mechanisms how these are regulated. And we thought, because I'm cell biologist, I thought that maybe neuronal activity will regulate the formation of new spines and maybe the initiation of the spines. So we started with the one protein which was in process, and this protein is called MIM, and what does it do? It curves the membranes. So there is first the flat membrane, plasma membrane, lipids, and then when MIM dimerizes next to the membrane, it curves the membrane. We did some live cell imaging, and we could see that this is now the GFP labeled MIM, and then we follow at the same time the actin filament visualizing protein. And we saw first that there's always, before new spine, there's first MIM clustering in one spot, and after a while there's coming out a new protrusion. If you know about the cancer cells, there are many protrusions in neurons. In principle, the principles are same. So these are philopodia looking structures in the beginning. After MIM is really out already, there's also filamentous actin is visualized. And this was kind of surprising for us because we thought that actin polymerization is very practical for the cells, because with polymerization you can push the membranes out and make all kind of cell changes and also maintain the cell structure. So actin is very crucial, but for our surprise, the MIM was enough for the first protrusion. And this we actually showed so that we inhibited the actin polymerization, and then we just got these small dots on the membrane. So MIM was possible to make the first curvature, but it didn't push the whole thing out on its own. We also did the overexpression experiments, so we have too much MIM in the cells, and then we see too many dendritic spines, proposing that MIM can facilitate the spine formation. We have the MIM knockout mouse, and we could show that these cells from the knockout mice, they have less spines. So MIM was also needed for the normal spine density. This we also showed in vivo. So we had mice, knockout mice, and we took from the cerebellum and visualized cells called Purkinje cells, where the MIM was highly expressed, and we could show that they have less spines.
I'm sorry to say 5 minutes.
5 minutes, good.
Or less, yeah.
Good. We checked also other molecular details. We know now that phosphatidylinoinositol 4,5-bisphosphate is needed for the MIM localization. We know that there is actin polymerization needed for this protrusion, and there is for specialists, it's up to 3 complex-based actin polymerization. And then we finalized these studies with studying the neuronal functionality, and we could see that the MIM knockout neurons, they were not working normally. And we also saw these mice that they have problems in motor coordination. So this was a nice example where we could show the combine from the very basic molecular mechanisms up to behavior, and these are all well linked together, which is not always the case. We continued with the behavior. Of course, the question is MIM needed for the learning? And we could show with now I'm going backwards. With 2 experiments, take it short, we could show problems in learning. However, this is not so easy. Because we also wanted to test in cells if the MIM actually react to the neuronal activation. This was not the case. We activated neurons, and in cells, we didn't see any reaction. So MIM is maybe needed for the learning, but clearly it doesn't react to the activation by itself. We also know that MIM is actually very developmental regulated, so it's expressed in most of the brain areas only in the early development, and in some areas only for the later life. So we wanted to search for new spine initiation factors which maybe would react for the neuron activation. And we had some candidates, and I jumped directly to the Gas7. And this is now Pushpa's work. I go forward. And this is now the video with the Gas7, and you can follow these protrusions, how they come in and out. And you can follow similar to MIM, you can follow here, there's a first little bit Gas7, then there's more, and protrusion come out. All right. Then we did similar experiments than for the MIM. So there is more spines if we have the overexpression of the Gas7 GFP compared to GFP. This is the analysis of that. We always make a computer-based analysis of everything what we can. And then we also check the expression, and Gas7 is expressed throughout the life in brain in many areas. We also checked if we downregulate the Gas7, then we can see that there is less spines in these neurons. We know that it co-localizes with actin, and clearly Gas7 can induce the actin polymerization. So in principle, some similarities to the MIM. We also know that there's NWASP in between the Gas7 and actin. Let's skip these up to 3 as well. And then we also know there's a PI345 trisphosphate involved. And then the last thing is that the activation actually neuronal activation can relocate Gas7. This is the first situation, and after adding the picocooline, which can inhibit the inhibition and activate the excitatory neurons, we can see the translocation to the membrane. And this is even nicer with the organotypic slices, so we need to make slices from the brain, and then we can infect cells, and we can show that here the Gas7 is as small dots, whereas after neuronal activation, the clusters come bigger. And this seems to facilitate the spine initiation. So with these experiments, we can link the neuronal activity to the changes in protein localization, and which will then facilitate the new spine initiation, leading to new connections between neurons. Okay, this is quickly the last summary. So we have now 2 pathways. Neuronal activation will induce PI3 kinase activation. We get PIP3, Gas7 is recruited to the membrane, and it makes this kind of broad structures to the plasma membrane, and this will end up to be a new spines. MIM is recruited with the PIP through. It makes a little bit different kind of protrusions. They are smaller, tighter, and there's a weaker link to the actin, but in the end, both of them induce new spines. And I stop here. You get still the snow view here.
Thank you very much. I'm sorry to interrupt you, because
No
the
worries.
time is very short. So can you introduce our second speaker?
Yes. Okay, so our second speaker will be online, and he is Professor Martin Goethe from the University of Münster, where I did actually my PhD, Germany. So welcome, Martin, online.
Time is 17 minutes.
7.
We tell him that the time is short.
I don't know. I think it's recording.
No sound. He starts to speak and no sound. He starts to speak and no sound here. Can you help? Yeah.
Martin, please, Martin, wait. Okay, can you speak now?
Hello, ladies and gentlemen. It's a pleasure for me to present my data today, and I would like to thank the organizers for inviting me to this great international conference. So today I will talk about Syndicum-1 as a pathogenesis factor and therapeutic target in breast cancer. And as you will see, this is a close collaboration between the department in Germany and Egyptian scientists in Cairo. Syndicum-1 is a transmembrane heparan sulfate protein glycan. Its extracellular domains contain heparan sulfate chains, which are carbohydrates that are similar to the pharmaceutic heparin. Heparan sulfate interacts with growth factors, chemokines, proteases, heparinase, and matrix proteins. So if you look at all the ligands that can bind to heparan sulfate under physiological condition, it's a big group of proteins. You can see molecules interacting like cell adhesion molecules, chemokines, cytokines, tissue remodeling factors, various extracellular matrix molecules, lots of growth factors, and factors involved in coagulation and angiogenesis. Apart from the heparan sulfate chain, syndicans have integrin binding domains in their extracellular domains, and they have highly conserved transmembrane and cytoplasmic domains. And these cytoplasmic domains are able to bind to elements which provide link to the cytoskeleton, for example, large PDC domain proteins such as syntentin. Syndicans have multiple physiological functions. They can act as co-receptors for insoluble ligands, such as fibronectin or collagens, and they often interact with integrins. They are also co-receptors for soluble ligands, and this is a very important function that I will come back to later. For example, they are co-receptors for many growth factors and their receptor tyrosine kinases. Syndicans can also act as endocytosis receptors, and an interesting feature is the shedding of syndicans. By this way, the extracellular domain can be cleave metalloproteinases, and this the membrane molecule to a soluble prime factor. So let's come to the classical co-receptor role. One, which is expressed in epithelial cells, can act as co-receptor for fibrous growth factor signaling. This is shown in this experiment. CF7 cells, the knockdown of CYP1 leads to a reduction of the split response to FGF mediated through the signaling. Cytokines can also act as co-receptors for deep protein receptors, such as pine receptors, and as we have shown, cytokines can also modulate notch signaling and co-regulate by the same proteins that activate. Considering these multiple functions, it's not a surprise that consider tumor progression. Some co-receptor role of factors can have an effect on the function of cell proliferation and growth. Moreover, a link to metastasis is there through modulation with proteinase and RNase activity. Local cell migration may be modulated as cell matrix interactions by an effect of silicon on stability. Our data furthermore suggests that there may be involved and exacerbation in metastasis. So what is known about silicon-1 in various cancers? It's been shown that silicon is frequently dysregulated cancer, but it depends on the type of cancer. Like cancer, it's silicon associated with a more aggressive phenotype. However, breast cancer, high expression is associated with focus and aggressive phenotype. So let's come to breast cancer. Breast cancer is the most malignancy, and it has been estimated that every 9 women is affected by breast cancer during a lifespan. Or every 20 women have breast cancer. What is known about silicon? We have shown that silicon expressed to the response to neoadjuvant chemotherapy. Moreover, we have shown that the signal of silicon-1 equal social factors of and genesis in early stages of breast cancer. Moreover, we could define a differential role for found in soluble silicon breast cancer progression, and we have shown silicon is regulated by RNAs, and its regulation promotes cell motility and effectiveness of breast cancer cells. Moreover, we study silicon-1 and resistance to irradiation in breast cancer. Hopefully, the group of Caroline Alexander in Wisconsin has shown that silicon not mice are resistant to breast cancer, and this reduced Wnt-responsive mammary gland precursor. So, these findings profit us to a hypothesis silicon-1 has breast cancer stem cell function. And this was investigated Sherry Ibrahim from Cairo, who was PhD in my lab and is now an independent investigator. What are cancer stem cells? Why is this a stem cells have a function generating differentiated cells. So there is stem cell niche, and they differentiate differentiation step into a defined tissue, such as cell, blood cell, or cell. However, if stem cells or the process are mutant, they can turn into cancer cells, which are very similar between stem cells and cancer cells. Stem cells are often more resistant to chemotherapy, the high expression of drug resistance. Moreover, there is DNA repair capacity, and this confers with radiation. Further, an unproliferative is controlled by the activation of polymerase which affect both. So I will present public work of cancer cells published with one radiotherapy resistance. Very early, we had investigated and analyzed what's the impact of Cynic-1 on the therapy, and it turned that in Cynic-positive patients, it was positive for Cynic-1, pathological response was decreased, and most Cynic-1 positive patients in the pulse were complete remission. So this suggests that Cynic-1 be associated with therapeutic resistance, and this could be due to phenomenon. So therefore, we investigate Cynic-1 function via siRNA down breast cancer cell lines. Sharif Ibrahim showed that Cynic depletion reduces stem cell expression, such as propagation, aldehyde dehydrogenase activity, and it also functional reduced inflammation of cells and in vitro. And Sharif could delineate that this Cynic-1 stem cell properties was due to downregulation in 6 downregulation of NRP and downregulation of co-receptor LRP. Return to Cairo, we continued our collaboration with cancer. And this inflammatory is very aggressive cancer with inflammatory tumor emboli. These tumor emboli express stem cell markers, but potentially to stem cell phenotype. What and co-workers found is that Cynic is higher inflammatory breast cancer, not IBC, and it correlates with the expression of the stem cell p44. Furthermore, show that notch expression relates to IBC, and function show he could show down of one cancer cell subpopulation inflammatory breast cancer. It's shown with different methods, including early colony formation, which is up on down, and also
I will do next.
silent leads pathways, which are also to compounds. The notch pathway were downregulated in cells, and also this was shown in the protein. So notch is activated by binding to on neighboring cells and plays an important role in establishing a cancer stem cell phenotype. Importantly, application of notch inhibitors in our in vitro to generate phenocopy of the Cynic silencing, meaning that it had the same effect as a Cynic and provides a functional link Cynic and the notch pathway. The working model for Cynic-1 function in IBC is that knockdown of Cynic-1 affects the 6 and the downregulation of the cancer stem cell population and to weakening of cancer properties. As cancer stem cells are, we ask the question: Cynic cells affect to and indeed, what us indicated that there may be a resistance to radiotherapy itself, and this is linked to activation of protein kinase signaling. And indeed, a group in Dresden, Germany, had shown increased integration leads to therapy resistance. So our experimental setup that we cells, transfected them to Cynic siRNA and irradiated cells, followed by functional assays. And indeed, Cynic knockdown increased resistance to irradiation in a conformation assay in 3 different breast cancer cells, which are triple negative. Moreover, the non-cynic infection led to an increase in breast cancer cell motility by video microscopy. So we next investigated the impact of spin knockdown radiation cell cycle. And what Teresa Rock could show in a laboratory that the knockdown of Seneca-1 under irradiation conditions led to an increase in cells in the G2 of the cell cycle, which is a proliferative effect. Moreover, she could show that the double-strand pair in radiation was put on Seneca-1 cells. We interpreted it as an improvement of the pair. We furthermore wanted to find out what molecular reasons for this resistance are, and perform transcriptomic analysis in combination with the University of Singapore. It turned out that gene relevant for cell separation apoptosis, for radiation associated repair, for cell motility, and radiation association signaling in related cells. And looking at this in more detail, Orhan Hassan in my lab could show there's increased activation of fertilization kinase in Seneca-1 depleted cells, and this may confirm resistance to radiation. Furthermore, analysis of qPCR showed that cyclins, most notably cyclin-dependent kinase 6, were downregulated in silicon-depleted cells. And this was also confirmed at protein level by Western blot. So therefore, our conclusions for progress are that radiation resistance of silicon-1 depleted cells is linked to altered DNA repair and enhanced invasive behavior. And this may be linked to integrin-related signaling pathways and cyclin-dependent kinases. So as CD44, the cancer stem cell marker and hyaluronan receptor, is downregulated in silicon-depleted cells, we reasoned that application of hyaluronan, which is a carbohydrate CD40, may be able to compensate for the loss of Seneca-1 breast cancer cells. This was published last year. So we tested the Seneca-9 and added hyaluronan to the system, hoping to make, let's say, for Seneca-1. However, we found that hyaluronic acid only has a moderate modulatory effect on the Seneca-1 dependent cancer stem cell phenotype. Only in MCF7 cells we could see a compensatory effect in a hanging drop acid. But furthermore, we could see a compensatory upregulation of hyaluronan synthase, the downregulation of another hyaluronan receptor Rna. So for the stem cell phenotype, this was less relevant. Stronger effects were seen on the cell cycle doses.
Sorry, Dr. Martin, we have 3 minutes left.
Finally, we want. Furthermore, finally, we wanted to see if there's a specific role of heparin sulfate modifications in cancer stem cells, because we had previously shown that enzymes modifying heparin sulfate leads to change in Wnt signaling compounds such as TCF4 in breast cancer cells. And we also showed that heparin sulfate transferase also Wnt signaling. When we looked at sulfate transferase overexpressing cells, but the spherical size may be genius. Furthermore, when we look at signaling, a very complex picture emerged, and we can say that there is an upregulation of not signaling compounds, but otherwise picture very context dependent. So to summarize, we could show that Seneca-1 is a positive regulator of stem function. Hyaluronan affects the Seneca phenotype slightly. Specific patterns of heparin sulfate enhance the cancer stem cell phenotype in a complex manner. Notch signaling are all heparin sulfate in this context, and the alteration in aberrant sulfates reach highly context-dependent effects. Nevertheless, we conclude that syndicamide defined aberrant sulfate structures emerge as pharmacological targets of breast cancer stem cells. And with this, I would like to thank the people who did the work, my PhD students and my collaborator, Sherif Ibrahim, and also my student, Nora and other PhD students. And of course, the funding organizations, EFG, DAD, the project Blindness. And with this, I would like to give you attention.
Thank you very much, Martin, for the nice results and sharing them. Next, we go directly to the second online talk, and this will be given by Professor Dr. Chandan, and he is Associate Vice President of Military and Applied Research in Indian University in USA. And his title is Tissue Nanotransfection Technology for In Vivo Tissue Reprogramming in Regenerative Medicine. Thank you.
Can you see my slides? Hello, can you see my slides?
They are not yet on the screen, but should.
Hello?
They are soon on screen.
Can anybody hear me? Hello? Can you hear me?
We can hear you.
Okay, thank you. Can you see my slides? Can you see my slides?
Not yet. They are not on the screen. They are ready to come, but they are not yet on the wall.
Yes, it's appear now.
But now they are. Please, let's go.
All right, thank you very much, and many thanks to the organizers for this opportunity to present. I am Chandan Sen from Indiana University School of Medicine and also affiliated with Purdue University Weldon School of Biomedical Engineering. Greetings from the US. So this is, in case you want to find us, this is the Indiana Center for Regenerative Medicine and Engineering. And you can Google for it using IUICRME, and you can find me at www.chandansen.org as shown on the screen. So I'm going to talk about a particular technology, and this is sort of late breaking, and my goal here would be to sort of summarize, given the time constraint we have. I'll not have the time to get into specific details, but an overall summary, assuming that most of you are not introduced to this technology. Now, regenerative medicine is, as we all know, an unfolding field, and things are getting started. The FDA has started a new track. Normally, there are drugs and devices, but now there is also something else in the US FDA called the RMAT, Regenerative Medicine Advanced Therapies. And in this, you have primarily a whole number of cell and tissue-based therapies and other types of therapies that align with the regenerative medicine theme. Now, one of the key concerns in these types of cell and tissue-based therapies is the fact that there could be certain unforeseen and foreseen adverse effects. So the FDA is particularly concerned about those adverse effects. Now, before getting into those adverse effects, how we are trying to, address them, and why are we approaching the topic of in vivo tissue reprogramming, let me give you a little bit of a foundation. So we all know that when we are in the fetal form, tissue plasticity is very high, as shown on the screen. When we are born, this tissue plasticity is somewhat blunted, but then when we are adult, it further pretty much is lost, because we were taught at that time when we went to school that these tissues got terminally differentiated, and therefore they lost pretty much all plasticity. But if we fast forward, a few years, what we will find is that fetal plasticity is high. Of course, that continues to be the case. In child, the plasticity compared to fetal goes down a little bit. I'm not talking about a cell. I'm talking about the tissue as a whole. Okay. And in adults, we are moving away from the concept of terminal differentiation to the fact that within the adult, there could be certain tissue that have really high plasticity, as seen to the right. In the center, you see tissue with plasticity that is intermediary, and some tissue have low plasticity, but there is hardly any tissue that has no plasticity. So the good news here is that adult tissue does have plasticity that we can play with, depending on what we would like to achieve. That, if we now go to the fundamentals of stem cells, so stem cells obviously have high plasticity, and that's why we are so excited about them. From stem cells, we can get more stem cells, and we can get a number of specialized cells from these stem cells. That is why we are so excited. But what we typically don't recognize that often is the fact that among the specialized cells, we could also end up getting cancer cells. So the idea of playing around with stem cell is a very attractive one. I think it has a tremendous potential, but to keep it safe is not trivial. It will take a lot of effort, because when we move stem cells into a given fate, a given specialized cell fate, not all stem listen to us. Some of them listen, some don't. And where you have residual plasticity or residual stemness, you face the prospect of maybe making cancer cells. From a regulatory standpoint, that's not okay. In other words, when we are treating somebody, we could either help them or maybe fail to help them. We cannot hurt them. We cannot hurt them. So there's a certain amount of risk associated with the use of stem cells or cells that have residual stemness. That said, we all know the massive field of IPS. A large number of work is going on at this time. So what you do in IPS is you take cells, it could be your autologous cells or not, but even if they're autologous cells, they are many weeks in the laboratory. And they're treated with reprogramming factors. You wait a few weeks, then you make stem cells out of them. Say for example, if you started with, say, skin fibroblasts, you make stem cells out of them. And then after you make stem cells, you then find a way to take them to specific cell fate that we are interested in. This is many weeks of work in the lab. And even if these are cells coming from our own body, when these cells come back to our body, in many cases, the immune system objects. And also here, we must pay attention to the fact that not all IP cells will listen to us and become the specialized cells we want. Some cells will not become the cells we want. We don't talk about them in the manuscripts very often, but those cells are there to do whatever they want. And, God forbid, they make cancer cells. That's then a big no-no for the entire treatment regimen. So that said, if you sort of summarize that, you have, if you talk about embryonic stem cells or any, you have neural stem cell, mesenchymal stem cell. Of course, you can talk about adult stem cells, adipose stem cells. or so, as an example. So from there, you can differentiate and you can have these types of specialized cells that you want. But then please pay attention to the bottom of the slide where it says direct conversion. So then the field of direct conversion came, in vitro it came, showing that actually a fibroblast, in order to become a neuron or a neural-like cell, does not have to go through any stem cell intermediary. That's called direct conversion. So in this case, you have these three transcription factors, BAM, and using that without going through intermediary stem cells, you can directly go into the cell that you want. What is the good news? The good news is you don't have any intermediary cell with residual stemness hanging out and then perhaps giving us a bad fate like cancer. So this whole idea of direct conversion, which was reported in vitro, got us very excited. And we wanted to achieve something like this, not cell reprogramming, but tissue reprogramming, not in vitro, but in vivo. So what we are talking about is tissue reprogramming in vivo. So basically, our goal was to develop a technology to reprogram within the live body. You need no laboratory, okay? And when you do that, you do the reprogramming under immune surveillance, meaning even if you take my own cells and you take them to the laboratory, you don't have my immune system there. So in the body, when you reprogram and you don't take them to the lab, then constantly the process is under the surveillance of immune system. So when that is the case, then whatever reprogramming process is happening in the body is constantly negotiating with the immune system and other systems that are in vivo, in that microenvironment, or even the whole system. And if you are able to achieve reprogramming under those conditions, then we believe you would have sustainable functional impact. Not to speak of the fact, by the way, you just got this done, making the body a laboratory. You don't need a specialized, GMP type laboratory. So scalability of the technology then becomes very, very wide. You can actually get this done at a very low cost all throughout the world. You don't need sophisticated laboratories. That is sort of secondary. What is primary here is the fact that you're not transplanting any cell into the body from the laboratory. These are tissues that are changing their fate or state within the live body. So with that said, we come to one of the early pictures, that we sort of reported in 2017, a few years ago now, 5 years ago. So what we came up with a particular nanotechnology-based silicon device, a chip as we call them. So in this case, you place the chip on the skin. So here, you remember I was talking about the previous slide, that in the adult body, you have some tissue that have high plasticity, some tissue that have intermediary plasticity, and some tissue that have low plasticity. Good news is, among the tissue that have high plasticity is the skin. So what you do this here is you take this nano device, you place that on the skin, and then you pass these pulses, following which these reprogramming factors that are on the device are electrophoretically pushed into the tissue. Now, a lot of people try to do this in the literature today using virus, which is fine as it relates to experiments, but you don't want to have reprogramming factors mounted on a virus, because then it would go into the body and could end up indiscriminately infecting tissue that you don't want to reprogram. And that is a big risk. So we counted not on virus, but on a biophysical approach, in this case, a Nano Electroporation approach. Why Nano Electroporation? That's a long story. I can answer the questions, but anyway, a very long story, very short. The device stays on your skin less than 1 second. I repeat, the device stays on your skin less than 1 second. The procedure altogether is 100 milliseconds. So the device does not stay on the skin, planted or something. It goes away after it touches you. All right. So the first paper came out a few years ago, like I said, 2017. After there, until now, there are about 6 to 8 papers that are out that talk about different aspects of this technology. This technology, for those of you that may be interested, has been reviewed in JAMA, has been reviewed in Science Translational Medicine, and also recently there has been a blog by the NIH director, Francis Collins, and the first author in our paper was a postdoc in our lab, also received the NIH Director's Award on this technology. So if you were to find out, I think you will find more than 500 videos on this technology in YouTube. So just go write tissue nanotransfection, and everybody has an opinion about this. So it looks like there's broad interest, and I hope a number of laboratories pick on this and run with this. I'll show you the versatility of this program. So here's a small video that I want to share with you. Can you listen to the sound?
As cells differentiate, they acquire specialized functions. For decades, scientists believed this process ended with terminally differentiated cells. Committed to a particular cell fate, the ability to switch function irreversibly lost. In a major breakthrough, iPSC technology established the possibility of inducing plasticity to terminally differentiated cells. But several limitations arose, including a lengthy multi-step lab-based process, possible inefficiencies due to experimental conditions, low fidelity with which the final cell type is generated, and safety concerns, including potential cancer risk. More recently, scientists found that mature, fully differentiated somatic cells could be directly converted to another cell type by ectopic expression of specific transcription factors, without the need to acquire intermediate pluripotency. However, this technique requires viral gene transfer, an approach which, 1, requires an exhaustive laboratory procedure, and 2, carries risks, such as insertional mutagenesis, irreversibility caused by stable transduction, and increased cancer risk caused by long-term proto-oncogene expression. Therefore, it is not well suited for point-of-care applications. Thus, a non-viral tissue nanotransfection approach was developed for direct cell conversion in vivo by Professor Chandan K. Sen in collaboration with Professor James Lee. This work has been published in Nature Nanotechnology in 2017. This approach is simple, does not require elaborate laboratory procedures, and can be used as a point-of-care approach. This approach consists of 2 components. First, a hardware component, or chip, fabricated for efficient delivery of cell-specific cargo in murine model. Manufacturing of this chip has been scaled up to fit dimensions relevant to human need. Such chip has been successfully tested in preclinical porcine studies. Second, the cargo itself, which consists of a combination of different reprogramming factors. Thus, by applying a highly intense and focused electric field through a array of nanochanels, this device nanoperates the adjacent tissue cell membranes and electrophoretically drives reprogramming factors into the cells. This new Nano Electroporation technology is distinctly different from bulk electroporation. The simplicity and utility of this approach was demonstrated by Nano electroporating the skin that served as an agricultural land to cultivate cells of different functional abilities. Using a specific cargo, a combination of different reprogramming factors, skin fibroblasts were successfully converted to endothelial cells that ultimately formed blood vessels with 7 days. These blood vessel networks anastomosed with the parent circulatory system and restored tissue and lymph perfusion in 2 murine models of injury-induced ischemia. Similarly, using a different cargo, skin fibroblasts were converted to neuronal cells that showed similar gene expression profile and electrophysiological function to that of cells in the developing brain. This tissue nanotransfection platform technology has the potential to ultimately enable the use of patients' own tissue as a prolific immunosurveilled bioreactor to produce autologous cells that can resolve conditions locally or distally upon harvesting. This simple-to-implement nano electroporation technology generates favorable biological responses. It is a one-time treatment applied topically and only lasts for a fraction of a second, without elaborate laboratory procedures. And it could have applications beyond reprogramming-based cell therapies, such as gene, protein, and drug delivery.
I hope you could hear that video. If not, you can also find the same video posted in YouTube. So please, depending on your interest. So after we published this, there was a lot of interest, obviously, that was asking us for the chip. And, we could only make so many. It's a, time-intensive, effort-intensive process. But more recently, What we have done is we have taken this chip, this nanofabrication process, to manufacturing grade. And that is currently published. So what you see here are electron microscopic images of the chip. The paper just got published. It is in PubMed and Nature Protocols. And I will spare you the description of all of this. These are some of the descriptions of the nanochannels. We call it the TNT chip 2.0. And we also, based on that chip, recently did some simulation studies, which are currently in press of nano research. So the simulation will tell you, once you use the chip, how are the reprogramming factors delivered electrophoretically, at what depth, and how can you control them. With that said, I'll take to another quick video before I close, which would be showing about the fabrication of this chip, should you be interested in how the chip is made.
Certain organs in our body can serve as a bioreactor. Through real-time feedback from our immune system, we can grow new body parts. We can even grow them in one place and then transplant them to where they are needed. One well-suited organ for such bioreactor function is the skin. This largest organ of the body provides ample real estate to grow therapeutic functional tissue structures. Tissue nanotransfection technology, or TNT, enables in vivo reprogramming of the skin to produce functioning blood vessels, active nerve tissue, insulogenic tissue, and more. This process depends on specialized chips created from silicon wafers. A coating is added to the top, and lasers are used to write small holes into the chip, then etched deep into the wafer. On the other side, a new coating is added and then lithographed to create hollow microneedles. The chip is then etched to make the needles longer, cleaned, and then treated with a new coating to improve precision. The exact nanofabrication process for manufacturing the TNT chip is now reported in a new publication in Nature Protocol. The delivery mechanism consists of two components. First, the chip, fabricated for efficient delivery of cell-specific cargo. Second, the cargo itself, which consists of a combination of different reprogramming factors. The cargo is different for different applications. By applying a highly intense and focused electric field through arrayed nanochannels, this device then operates the abutting tissue cell membranes and electrophoretically drives reprogramming factors into the tissue. In this approach, the skin may be viewed as an agricultural land that can be used to cultivate cells and multicellular structures of different functional abilities, such as perfused blood vessels, electrophysiologically active nerve tissue, etc. Using a specific cargo, a combination of different reprogramming factors, skin fibroblasts were successfully converted to vasculogenic cells that ultimately formed perfused blood vessels within 7 days. These blood vessel networks anastomosed with the parent circulatory system and rescued against ischemic insults. Similarly, using a different cargo, neurogenic as well as neurotropic reprogramming skin cells were achieved. Such intervention proves to be promising to rescue against stroke as well as diabetic neuropathy. In future, TNT is expected to involve dispensable chips with injectable factors supplied as liquid-loaded cartridges. During use, the chips will be stamped on the skin. Such approach will be compatible with point of care using minimum training for local and remote use. This TNT platform technology has the potential to ultimately enable the use of the patient's own tissue as a prolific immunosurveilled bioreactor to produce tissue, grow body parts, and resolve conditions. It is a one-time treatment applied topically, only lasts for a fraction of a second, and can be applied at point of care without elaborate laboratory procedures. And it could have applications beyond reprogramming-based cell therapies, such as gene therapy, protein delivery, drug delivery, and cell-specific exosome labeling.
Thank you. That takes me to my final slide in the interest of time, and which is the number of stuff that we have so far enabled. To your left, you see the vasculature. That's what is published, meaning that you take say a hind limb ischemia model, and you do this procedure once. In 7 days, when in the case where the femoral is completely transected, you can rescue that entire leg using TNT only once. And you start seeing those vessels from day 7 onwards, and that is already in the literature. The second, I think the first paper is in press on that, is, in the situation of lymphedema, where you need, today, what surgeons do is they surgically anastomose the lymphatic vessel with the vein. So this is called lymphovenous anastomosis. With TNT, we have been able to come up with an approach that can actually cause lymphovenous anastomosis. You don't need a surgical procedure, and you can get relief from lymphedema. The third example that you see here is unpublished, and actually the first experiments on primates just happened last week.
Sorry,
yes.
Dr. Chandon. Time is really up. We need to be very strict with the time. Can you just conclude and then?
I just said this is the last slide.
Good, thank you.
So the third example is, these are within the cells that you can make beta-like structures that make insulin, and that releases insulin into the diabetic. And this release of insulin is guided by the level of glucose in the blood. The next example you see that we have published on is that you can improve diabetic wound healing by bringing in vasculature, because diabetic wound healing has a vasculopathy and neuropathy with it. And the neuropathy example has been published. We have been able to also use this in solid tumors. This paper is currently submitted and not yet published. And you can regress tumors using TNT. We have also published in ACS Nano, the journal ACS Nano, that you can use TNT to cell-specifically tag, fluorescently tag exosomes. So when you see exosomes in the blood, you can tell which organ it is coming from. We have already published that you can make induced neurons using TNT, and there is a paper soon to be coming out that is currently in revision that has achieved gene editing, in vivo gene editing using TNT. With that, I would like to thank you for your attention. Thank you.
Thank you very much, and we will proceed to our next speaker, Professor Dr. Wael Abdul Khair, Professor of Stem Cells and Immunology, Cairo University. He will talk about stem cell research and medical applications. Thank you very much. Sorry, Professor Wael, the time is very limited. So I'm sorry, I will interrupt you at the end of the timing. Okay.
هو البوينت انا مش لاقيه حد من الدكاتره اخده عضو ده مثلا هتقبل من بره البوينت نفسه من بره لا ده بتاع هو ماشي بص هاي انا هاخد لك نص ساعه على تلت ساعه ماشي هيرن لك على تلت ساعه اعمل ايه؟ هيرن عند تلت ساعه تمام فتحت المايك مفتوح المايك ايه فتحت زرار صوت تمام
بسم الله الرحمن الرحيم. I want to thank Professor Dr. Hussein Sabit for his bring me to this big conference and as a good organization, and I thank all the organizers for this big conference and international impressive organization. Now we will talk about cell-based therapy as a hope for incurable diseases. Stem cell, what they are and what they do. Stem cells are primitive cells found in all multicellular organisms, characterized by self-renewal and the capacity to differentiate into any nature into any mature cell type, as Dr. Shendon say in the previous presentation. These stem cells have the awesome potential for regeneration and may be used to replace or repair damaged cells. This image, as Dr. Shendon say, it is here is a stem cell which replicate, giving another stem cell, which is a renewal of this cell and give specialized cells also. Why is such an interest in stem cells? Regenerative medicine promotes the repair response of disease, dysfunction, or injured tissue using stem cells or their derivatives. It is the next chapter in organ transplantation and uses cells instead of donor organs, which is limited in supply. It also hopes for incurable degenerative diseases. Also, in some cases, instead of undergoing extraordinary expensive drug treatment, the patient would be able to utilize stem cell therapy, and the patient would avoid chronic pharmacological treatment and its inevitable side effect, as in autoimmune diseases. The patient must use either cortisone or cytotoxic drugs. What is this opposition to stem cell? When we hear about stem cell therapy, we are fair and say it is a fake and it is not true. So many people say that it is realms of quackery, شعوذة يعني. Embryonic stem cell remain controversial because of their derivation from killing early embryo. وده طبعا ما بيحصلش. It is not true because it is not from the embryo, it is from the blastocyst, which is not used after in IVF. Other stem cell cannot be changed into all different cell tissues, but we will show in the next slides that mesenchymal stem cells are multipotent and mesenchymal stem cells are an adult stem cell. Other stem cell from a person's bone marrow can be extremely painful, but it is taken under local or general anesthesia, and all procedure in medicine may be painful. Stem cell research is costly process, but it is less costly than surgery and tissue transplantation. We don't know if there are long-term side effects to worry about. Since 60 years, tens of thousands of people are receiving stem cell transplant every year for blood diseases as leukemia and myeloma, with efficacy rate improving without side effect and without causing carcinoma or malignancy, but it treats carcinoma and the malignancy. Stem cell therapy have no evidence to back up those claims. Thousands of evidence-based published literature we will say now. Sketchy clinics, they say, sketchy clinics pull people in with big promises and slack advertising anecdotal testimonials posted in online videos, on blogs or social media. But all medical staff in many specialties doing that with the case presentation of their treatment. They say also some mimic more reputable scientific endeavors by posting their work on clinicaltrials.gov, a federal respiratory that doesn't always vet posting to ensure their protocol meet federal standards. But the USA National Institute of Health is the most specific scientific reviewer in the world. It is not a third world countries to say that. It is a powerful scientific site. If you search in PubMed or National Library of Medicine in Center of Biotechnology information about safety of stem cell therapy, it is many literature published, about 9,000, and it is increased interesting in this publication after in the last years until now. Also, we found about 147,000 and more if we search about stem cell therapy in NIH or PubMed.gov. And also it is the result increased by the years. And we found about more than 16 stem cell therapy, 16,000 stem cell therapy clinical trial on literature, published literature, with evidence-based study. In clinicaltrials.gov, which is the US National Library of Medicine, we found about more than 6,000 studies found. Clinical trials which done on patients. This is a map showing the distribution of the stem cell clinical trials and researches. Clinical trials, these are clinical trials all over the world, showing that it is more in Europe. It is more in Germany and France and Italy and USA. And this is also in Middle East and Egypt and other countries. These types of stem cell, either embryonic stem cell, which taken from the, excuse me, a pointer for you, because it is not work. This fertilized over in the day 6 or 7, we take in the inner cell mass, which containing the embryonic stem cell. The reprogramming cell induced the pluripotent stem cell, which Professor Dr. Shandon in the previous presentation speak about it. There's many methods of reprogramming of the cells and induced multi-totipotent stem cell or pluripotent stem cell. Also, we take stem cell from adult tissue as blood, bone marrow, and adipose tissue, and this giving multipotent stem cell which can transfer differentiate into neural cell, beta cell, of islet beta cell, blood cells, hepatocyte, cardiac cells, and others. The cells are either totipotent, which is the fertilized egg, and after 5 to 7 days, it is embryonic stem cell, which is pluripotent. And after that, it is adult stem cell, either cord blood or placental blood or cord wort jelly. And from the bone marrow, it is a multipotent stem cell. Multipotent, which give a variety of specific tissue by transfer differentiation. The potential uses of stem cell in many diseases, in neurological diseases, stroke, traumatic brain injury, learning defects, Alzheimer's diseases, Parkinson's, in dentist missing teeth, wound healing, and other many application, and in autoimmune diseases in rheumatoid arthritis, Crohn's disease, and multiple sclerosis, and others. This disorder treated by stem cell, progressive muscular, all of them is degenerative disease that don't, there's no hope for treatment of them. Progressive muscular dystrophy, spinal cord injury, cerebral palsy, ataxia, motor neuron disease, and femur head necrosis, and others. How does it work? In bone marrow transplantation, which FDA approved from 60 years, bone stem cell replace cell damage by chemotherapy to fight some types of cancer and blood-related diseases such as leukemia, lymphoma, multiple myeloma. This transplant use adult stem cell or umbilical cord blood stem cell. In autoimmune diseases, mesenchymal stem cell have been found to be effective in suppressing excessive immune responses such as graft versus host disease and systemic lupus erythematosus in both animals and humans. Stem cell use in dentistry. There is many application of stem cell industry in the dentistry, as in mandible bone defect. We're doing a graft with nano particles scaffold with a stem cell to elongate the mandible, or to elongate the mandible in malocclusion of the mandible, and in bone, in teeth, in dental implant, in bone resorption. Extravesical based therapies, which is the new treatment more effective than stem cell. It is one of mesenchymal stem cell product containing cytokines such as interleukin 6, 8, monocyte protective protein, vascular endothelial growth factor, platelet-derived growth factor, basic fibroblast growth factor, messenger RNAs, microRNAs, and non-coding RNAs, and several proteins. All of this, and especially the non-coding RNAs, create epigenetic regulators, which lead to the formation of epigenetic modification in resilient cell in congenital disease. This chart showing how stem cell or mesenchymal stem cell modulate the immune response by inhibit by these cytokines which I mentioned before, and it inhibit proliferation of B cells, T cell, natural killer cell, and dendritic cell, and the monocytes and macrophages. So it helps in autoimmune diseases powerfully more than the drugs that inhibit or destruct the immunity. It only regulate, it increases the T regulatory lymphocytes, the B regulatory cells, and the regulation of the immunity. This one of our animal studies on canine model for spinal cord injury. We doing cutting or crushing of the spinal cord to cause hind limb paralysis. As we're showing that there is hind limb paralysis, you can not move, and this is the control dogs. After injection of stem cell, we classify these canine models into intact canine model and control canine model, which we're doing transfection of the cord, and another injected by autologous bone marrow stem cell from the canine model. We stand again and can move. And after that, we harvest these animals and showing histological examination. We found that there is in the control dogs which are not injected by the stem cell, there is maceration and evacuation in the spinal cord, and the dogs which are injected by stem cell becoming regenerated spinal cord with a nucleus and no evacuation. After that, we're doing hematoxin and eosine and the immunohistochemistry staining, showing a maximum cavity area and showing the maceration here in the low power and high power. And the injected group, there is no evacuation, and the maximum cavity. The maximum cavity become smaller than the control. Also, we're showing the myelin-basing protein after injection of stem cell that increased in the curve, and the locomotor score, which and the frequency of hind limb movement in the animals. And then we were doing a multicolor immunofluorescence staining of stem cell of spinal cord injury sections from dog treated with autologous bone marrow stem cell stained with green fluorescent protein and doing multicolor for neurofilaments and the Debye stain, showing new myelination, intense green fluorescent protein staining, and the neurofilament was observed in the dorsal function, indicating peripheral myelination. Then it is published in the American Journal of Neuroprotection at 20 and 8. And after that, we also published another paper which is by staining with green fluorescent protein to track the cells in cell transplant and published in PubMed. This other presentation, if you want to know about our work in stem cell, you can go to Google Scholar to show about more than 50 work in our studies. Treatment instruction and the constant. The patient signs an informed consent that he seeks stem cell therapy and knows that it is in the process of research and that it is not approved by the ministers of health and that the results are not confirmed and that the patient will bear the cost of the consumables, treatment and post-treatment and knows the possible side effects. We'll show one presentation case for the time. This is a patient with spinal muscular atrophy.
ارفع لفوق كده.
ممكن نوطي الصوت لو سمحت عشان الصوت عالي قوي بتاع او نلغي الصوت خالص يعني. دي patient عندها spinal muscular atrophy coming to me when she complaining from unmanipulation. She had scoliosis, severe scoliosis and kyphosis. She can't sit without support from behind and from the side. She can walk, cannot elevate her arm, and cannot move her wrist or fingers. She had respiratory failure and the tongue protruded outside. We'll show the case.
ده اخرك؟
Cannot elevate the arm more than that.
اخرك كده. طيب كده بتسويتش.
There is deformity in the joint.
يعني هنا في deformity هنا اهوت ابتدى اللعق العظمي يمكن مع بعض. وهنا القدم برضه نفس الموضوع في deformity وهنا برضه.
From the long time of wheelchair using and don't unmanipulation, there is deformity in the joints, and there is unsupported neck. She can unsupport her neck. She cannot elevate her arm to take the napkins from me, and cannot move her wrist or fingers at all. There is any movement, no any movement before treatment. After 1 month of injection of stem cell, she can support her neck. She can sit with some support, and she can respirate freely, no tongue protrusion, and can move her legs. After that, we're doing another injection for 2 months. After 2 months, she can sit and without support, and I say to her that you supported her arm to the bellow, and she left it, and no support from behind or from beside, and she took that it must before injection must support herself by in the side and in the back. Also, she can check her knee, can respirate freely, also no respiratory distress, can move her wrist and fingers, can open the door, can eat, can change her clothes. The quality of life become good. It is not becoming a normal patient, but it is different, and she can moving, we'll see, by the wheelchair to the front and to the back. Now she can doing flexion and extension of the fingers, can extend her knee with the deformity, okay, but the muscles become powerful to move the joint. And I move the joint to show that before that I can't extend her knee or doing extension of her knee joint, but after injection by manual, by herself and by manual extension, I can't move her knee. So that we showing the hip joint movement. She can flex and flex and contract her knee. Flexion and extension of the hip joint. She can flex and extend and moving the knee joint and the hip.
Very impressive.
And she can elevate her back from the bed. Is now she can, after 4 months, she can doing that, moving with wheelchair. Her legs before injection cannot touch the foot, the floor, because it is a short ligament and no movement at all. This patient now becoming more and more better. Thank you
She
very much.
needs a continuous injection or
No?
she needs continuous injection with stem cells or
No,
this will be the
we inject about 5 times for 5 months, and she doing physiotherapy with our injection to, but she had this degenerative disease of spinal muscular atrophy, a congenital disease, that no treatment for these cases. And now the FDA approved an injection for spinal muscular atrophy for patients below 2 years. It is about $35,000 one injection, and the first patient taking this injection died. And this, it is very cheaper than that more time. Thank you.
congratulations. يعني it's ربنا يبارك لك.
Thank you very much.
Thank you very much. Then we go forward, and the last speaker of the session is Professor Dr. Anwar, and she will speak about genomic and public health.
هظبط المنبه على بس يبين حضرتك بعد 20 دقيقة.
انا هخلص على طول عشان اللي بعدي.
نعم 20 دقيقة.
سامعيني كده؟
الصوت واضح؟
ما انا
مفتوح.
فتحته.
طب معلش يا دكتور. جربي كده يا دكتور.
كده سامعين؟ لا اشتغل؟ ايوه مساء الخير. يعني احنا جايين في آخر الوقت. I hope you are still awake to listen. Actually, my talk was like an introductory talk or a concluding talk. It's about genomics and public health. My specialty is public health. So I was involved in biotechnology and genomics. Maybe some people are surprised why I am interested in this. Maybe what I will present may explain why. Okay. Of course, we hear about several disciplines in dealing with DNA and molecular technologies. We need to differentiate between genetics and genomics. They are very similar disciplines. They both focus on DNA of the organism, with a major difference being in their scope. يعني. Studying both genetics and genomics is important for different applications in medicine. Genetics, it's the study of a single gene and their effects. But genomic, it's a study of all of the genetic material belonging to the organism. Genetic research include molecular research done in the lab and ranges from gene therapy to gene editing using transgenic and engineered animal models of human disease, while genomics using high performance computing and math technology, known as bioinformatics. Genomics researcher analysis enormous يعني amount of DNA sequence data to find variation that affect health, disease or drug response. يبقى يعني genetics, it's a single gene study and its effect usually it's in the genetic diseases. In the clinical we see it. There is a classical genetics, but genomics is a study of the whole genome. It introduces a large يعني volume of data and needs a special way of analysis by bioinformatics. معلق ده. طبعا الجينetics يعني play an important role in understanding diseases caused by specific defective genes, while genomics require to have a complex problem of how the genome and the environment, for example, interact. As I said, it's the classical genetics. يعني for a long time, most of the genetic diseases were described by the phenotype of not the genome. When molecular techniques introduced, they start to know the mutations in genes and the details of to study these diseases. Molecular genetics, as يعني after all these lectures I attended from the morning till now, you are aware of all what I was trying to say here. And new technologies like CRISPR, and as we know about it, changed our mind towards this discipline. We heard about the diet, the genetically modified products, and gene editing in food products, and so on. So you are aware of it very well. And experts introduce new gene therapy approaches in muscle weakness, as we heard the stem cell, the last lecture. So the applications of genomics and genetic engineering is become broader now. We know the experts know it, and the application is a wide variety of things. But when we come to the ethics, why the regulations are difficult to reach and why the laws become, there are a lot of debates about it, because it needs to see it from the ethical point of view. Even in the plants, there are ethical way. In the animal experiments, there is ethical approvals. Of course, in human being, there are a lot of ethical discussions, and we have to have the ethical approval while we are doing anything. I hope that everybody, young staff who start to do research in DNA and this editing and so on, don't proceed except if you have the ethical approvals from your university or institution ethical committee. Especially in human, there should be an ethical approval. Even if you believe that you are doing a good thing for the patient, but you have to have his consent. If he is a child or not mentally aware, you should have the consent of the father or the parents or the relatives. So I was interested for a long time about the interaction between genetics and environment and its impact on health, and also the behavior of the human being towards these interactions. When we focus on health, because I'm talking about public health, we talk about the health of the individual, the health of the family, the health of the community, the health of the healthy environment surrounding us. And we are affected by different disciplines and technologies and science. We cannot separate. So there are several determinants that affect our health. This biomedical research or bioinformatics or genomic research and so on, it affects the health, not only the individual, but the whole community. That's why we're interested in. And there are multiplicity of scientific research that deal with this kind of studies. We have the molecular research, we have the health of the people, and there are community-based research. There are differences between having a system for the individual or for the community, the whole community. The good news is that there are several diseases now preventable. When we have all this kind of research, it is preventable. We should focus on the preventable diseases. And the genetic diagnosis, of course, started by the family history. If there is inheritance in these diseases, antenatal care is very important during the pregnancy to know if the child of the baby affected by certain disease, so we can avoid this. So we have the community-based studies, and we have the studies on genetics or the services provided for genetic diseases. We should look at it from different questions: when and where and how and how much. Who is doing it? When the genetic disease, it comes when the patient is in a late stage. But when we talk about community-based studies, it should be early detection for prevention of the diseases that I suggest to prevent. And where, of course, in the genetic diseases, it's in the specialized centers, in a specialized hospital, by specialized people. But the community-based can be done in the primary health care facilities. And this will include who? the primary health care doctor or the family health doctor can have a contribution in prevention of genetic diseases when he do early detection of the disease. Of course, I said the specialized genetic care group, they are the one who doing. Of course, the genetic diseases need counseling, need clinical, need laboratory, need facilities. And this one needs, but they focus on health education, to educate the mother, to educate the physician himself how to early detect the problem. How much, of course, the diagnosis and the treatment and the gene therapy and whatever is very expensive, but the community-based by early detection, the problem not coming yet, so it is less expensive. So I am encouraging now to focus on the primary health care facility, how they can have a role in prevention of these diseases. If the family doctor suspects any problem, he should refer to the expert who in the specialized centers. As I mentioned, bioinformatics is the way to analyze the big data we have. We have concerns, scientific, economic, social, ethical, talking about stem cells or cloning or whatever, genetic editing. These are, should be taken with cautious in several aspects. We have challenges. These new technologies should be seen by different experts, by different disciplines in order to have acceptable programs or so on, acceptable to the community. I want to go through this one. For the World Health Organization for several years are interested on this topic, on how genomics can benefit the world health, and not only public health of an individual or community, small community, but the whole world health. And all the time they put the top priorities in the technology to help. And I think the coronavirus or the COVID-19 showed how the importance of the genomics in diagnosis, in the vaccine development, and maybe regular physicians now know about RNA, about different technique to prepare the vaccine. So it's a really a big when it's a big problem, everybody talk about it and make it easy for understanding. As I said, ethical concern is very important. I want to say that as I said, I'm interested in the role of the interaction between environment and genetics. There is a field called epidemiology, genetic epidemiology, which in genetics in the community, and how these genetic problems is distributed, and there are several distributed among the people, place, and time, and how the risk factors affect, or the determinants affect this distribution. The environmental mutagenicity or molecular. This slide demonstrates how if we are exposed to certain agents, from exposure to the clinical effect, the cancer, for example, to happen, there are also a paradigm or a route from the introduction to the environmental agent, and then the metabolites appear in the blood or the urine or the body fluids, and then the starting of the changes in the DNA, and then the mutation that can happen the amplification, translocation, allele, يعني different changes happen inside the cells, and then the tumor appear. This diagram shows that at a time, maybe repair can happen. So if we stop the exposure, and this is the prevention, we need to diagnose the problem early. If there is a changes, we have to stop or to search for a cause to stop the process till the disease happen. So this is the interaction between different disciplines. يعني public health alone cannot see all the changes happening, and also the molecular people who are having this kind of research cannot see this root or diagram. So they have to work together to actually do a good study. This is what I'm saying. The top 10 technologies according to the WHO and so on, the molecular diagnosis, and we heard about different techniques today, and the recombinant vaccine, for example. Vaccine delivery system, also they introduced the bioremediation, also talking about using this technology for the environment and to remove the toxic waste and clear the environment from pollution. This is important also for the health. And the sequencing of pathogen genes. Yani, as I said, the COVID-19, without sequencing the virus itself, we couldn't make any progress in vaccine development and so on. Female-controlled sexually transmitted disease is one of the priorities. It's an old and continuing to be important. The bioinformatics is very, for a long time, World Health Organization is enforcing this discipline to be introduced for the sake of more research. The enriched GM crops and the combined drugs, combination chemistry. I want to say that in Egypt, molecular research is present in several places. We are here in this conference is organized by an important university, private university, but also research happened at different research centers related to the Ministry of Higher Education and Research. Ministry of Health, maybe they have in their central laboratory or in some hospitals, they do certain techniques. And Ministry of Agriculture, of course, private sector, universities and research institution, national program, Notkar can, Agiri, Vaxira. This is in different ministries. And of course, the Academy of Science, Scientific Research and Technology is interested. I would like to finish my talk by saying that there is a building blocks if we need to continue in these fields. We have to have several things. We have to have the leadership. We have to have the networking, the information sharing, the improved research culture, the programmatic use of research finding. I'm usually surprised by the large number of publications, and it's not used, this publication. We need to share the results of the publications in order to proceed in the next step. Expert saving, we search for the experts to collaborate together, the human resources, the challenges and rewards, the intimate cooperation, resource mobilization. Above and the top of the pyramid is the tailored strategy. I hope that the people working in science and in genomics and genetics to sit together to have a strategy for 5 years, and when they meet again in the conferences, they follow up on what they prepared for. Thank you very much, and I hope I summarized. Wallahi.
Thank you very much. This was very nice summary for the session, and then we have an announcement.
Okay. Thank you, Berta. Thank you, Professor Agida, for chairing this session. We are behind like 40 minutes of the scheduled time. Actually, I would like to ask you again just to compress the time of launch. I know the launch is a little bit far from here. It's in the playground there. So we will make our efforts just to minimize the time of launch because it's a box, so it will not take much more time. So please try to help me just to catch the time, because we need to finish at the time, because I know some guys from you, they are come from very far places. So again, I ask you to help me. Thank you again.
Sometimes when someone's saying something and then what answers promised.
أنا فادي أنا فادي. محمد ربيع تلاقيه هناك.
فين في اوضه المدير؟ أنا فادي أنا فادي سلامتك. ترند اقفل المايكات يا
احنا كله تمام
لا،
ولبسنا
تمام.
3 in advance وكله كذا يا حاج. هو ده اللي جاي ده هو بتاع
Good afternoon again. Good afternoon. Please be seated. Organizing committee, please be seated. We will take off. Lee. Yes. Dear respected audience, dear respected colleagues, please be seated. We will start this session. This session chairperson is Dr. Didier Monet. He will manage this session. We will start with the sponsor speaker, a talk for like 10 minutes from the company analysis. Just for some technical issues, we will have like half a minute, please.
Good afternoon. I'm hearing you, but the mic is not working.
We will introduce these guys.
Dr. Scholler? No.
We will take a sponsor talk for 10 minutes.
Okay, you are the
Analysis.
sponsor.
Yes, yes. Analysis.
Okay. I don't know your name.
Mohi.
Moin?
Just Mohi. Mohi.
Mohim.
Yeah.
Okay. Please, people who are outside can come inside. We are starting the afternoon session. I am Didier Monté from CIRAD in France, which is a research institute where I work for 40 years now. I am older than you. I don't know. What do you want to say? Because you are.
I'm just waiting for the presentation to be.
Yes. And it's
mainly about sequencing technologies and precision medicine.
Okay. Alors, we will have a sponsored talk session now, conference or presentation, given by Moin. It will be on sequencing analysis applied to medical science, if I understand well. It's okay.
سامعني كويس؟ مساء الخير، انا محيي ابو المكارم من شركه اناليسز. انا حاليا برودكت لاين مانجر سي اي اند ان جي اس انسترومنتس اند كيتس. النهارده هتكلم على السيكونسينج تكنولوجيز الافيلبل عندنا في البروفايل في الشركه عندنا. مينلي مش هخش ان ديبث في التكنولوجي نفسها، بس انا هقول وات از افيلبل وات وي كان دو. طبعا كل الريسيرشرز عامه سواء جونيور او سينيور ريسيرشرز او بي اتش دي هولدرز طبعا بيحتاجوا التولز اللي تساعدهم ان هو ينفذ الورك فلو اللي هو او الريسيرش ستادي اللي عايز يعملها. طيب شكلي في بروبلم في البوينتر. مينلي الطب عامة ابتدى بالابروتش انتويتيف ميديسن، ان انا بشوف شوية سيمبتومز بناء على السيمبتومز دي بحدد كيور معين. الكيور ده بيبقى افيلبل لكل البيشنتس with the same symptoms. ده كان في الفترة ما بين الثلاثينات للخمسينات، من الخمسينات من الثلاثينات للخمسينات. ثم بعد كده الموضوع اتطور من الانتويتيف ميديسن للايفيدنس بيزد ميديسن، where في الحته دي بنعمل كلينيكال ترايلز، وبيزد على السكسس بتاع الكلينيكال ترايل بيتم ريليسنج ل ميديسن، نوع ميديسن معين بناء عليه بيعالج الكوزز بتاعه المرض ده. الموضوع دلوقتي بعد 2010 الموضوع اختلف، بقى في ابروتش جديد اللي هو البريسيجن ميديسن. البريسيجن ميديسن يعني ايه؟ يعني كل individual is unique، الكونديشن بتاعته unique، سواء inherited about it's about inherited disease او certain disorder او نوع من انواع الكانسر، through which الريسيرشرز احتاجوا ان هو تولز معينه على طول السنين، من او بدايه من مندل لما لاحظ الفينوتايب بتاع البسله، ثم بعد كده الموضوع اتطور لسانجر اللي عمل بروتين سيكونسنج، ثم الدي ان اي سيكونسنج، عرفنا يعني ايه طفرات، يعني ايه جينات. ومع كل ده ابتدى بيبقى فيه تطور للتكنولوجيز الافيلابل اللي بتساعد الريسيرشر ان هو يعمل شغله. انا فاكر كان دكتوري في الجامعه بيتكلم ان هو الماجستير بتاعه كان على سيكونسنج اوف 1200 بيس بير، ده الماجستير بتاعه. حاليا عندنا انسترومنتس ممكن تعمل ال 1200 بيس بير in just 2 hours. بقى في ابروتش جديد اللي هو النكست جنريشن سيكونسنج اللي بيديني مور داتا، مور كريتيكال داتا بالنسبه لي، بيزد عليها بنبتدي ناخد ديسيجن كلينيكال ديسيجنز معينه. طيب احنا عندنا في شركه اناليسيز اللي هي وكيل ثيرمو فيشر في مصر عندنا 2 generations of sequencers، اللي هو الفيرست جنريشن كابيلاري الكتروفوريسز والنكست جنريشن سيكونسنج. الاثنين في النهايه بيصبوا في مصلحه الريسيرشر في ان هو بيديله تول معينه يقدر يجنريت بيها داتا، الداتا دي بياخد بيها ديسيجنز معينه او يابلاي ريسيرش معين في اي فيلد هو محتاجه، سواء الفيلد ده ميديكال او انفايرمنتال. الموضوع اتطور، ما بقاش بس فكره ان انا عندي كوزز معينه او سيمبتومز معينه بناء عليها بتعامل، لا هو كل انديفيدوال يونيك بالباترن بتاع الميوتيشن باترن بتاعه، اللي انا اقدر بيزد على الميوتيشن باترن ده اقدر اتعامل بطريقه معينه. فالموضوع بقى مش زي زمان ان انا بدور على ميوتيشن واحده بس، انا اقدر ان سنجل ران اوف ان جي اس اقدر اجنريت thousands of variants، يعني اعمل لها الاناليسيس بتاعتها، اشوف الكلينيكال سيجنيفيكانس بتاعها. التطور طبعا في البايو انفورماتكس دلوقتي عن زمان بقى رهيب. دلوقتي بقى عندنا بانلز بتكفر certain hematological diseases زي المايلويد covered fully with a certain panel بتكفر الجينات الموست susceptible مع المايلويد، ومش المايلويد بس، الليمفوما، الليكوب بايوبسي ريسيرش، السوليد تيومر ريسيرش، كل ده بقى متاح دلوقتي. وكل التولز دي بقت تساعدنا كريسيرشرز ان انا ابلاي البيست براكتسز واطلع بريزولتس، بناء على الريزولتس دي بقدر اساعد الانديفيدوالز ان هو يبقى عندهم هيلثي انف ان هو يوبريت طبعا، لان البابليك هيلث دي حاجه مهمه جدا لاي مجتمع. يمكن حتى فكره البريسيجن ميديسن انا شايف ان هو الاوقع، اللي هو البريسيجن ميديسن ان انا بعالج الكوز مش السيمبتومز، مش مجرد بس سيمبتومز. انا عندي كوزز معينه بتسبب المرض الفلاني او بتسبب لاك اوف بروتين فلاني او الانزيم المعين، فانا ساعتها بروح تو ذا بوينت ان انا احل المشكله دي. وفكره البريسيجن ميديسن ان هو مش جاست ان انا باخد جيناتيك انفورميشن، لا مش بس جيناتيك انفورميشن، ممكن اللايف ستايل نفسه يبقى فيه مشكله، ممكن الانفايرمنت نفسها يبقى فيها مشكله. يمكن حتى في امريكا في علاج الازمه زمان، الازماتيك بيبول كان هو علاج واحد هو انهيلر مع تابلتس معينه. الموضوع اختلف دلوقتي، بقى كل انديفيدوال بيزد على اللايف ستايل بتاعه، بيزد على الايج، بيزد على الجيناتيك فايندنجز اللي بيلاقيها الكلينيشن، بيقدر ياخد ديسيجن سوتابل فور ذس انديفيدوال. وده يمكن الابروتش اللي احنا داخلين عليه الفتره دي، او اوريدي هم بره ابتدوا يشتغلوا بيه من فتره، واحنا حاليا في مصر ابتدينا نجو فور ات من خلال مشروع الجينوم المصري اللي حاليا بيتم تنفيذه في مصر. بشكل عام اللي انا كنت عايز اقوله في البرزنتيشن ان هنا عندنا التولز اللي تقدر تساعدك كريسيرشر ان انت تنفذ الجولز اللي انت محتاجها من الريسيرش ستادي بتاعك، بلس ان احنا نقدر نعمل نوليدج ترانسفير مع الوكيل اللي هو ثيرمو فيشر، الفايندنجز اللي بيلاقوها، البيبرز اللي بتتنشر، تساعدنا برضو ان احنا يبقى عندنا الانتيتي بتاعتنا كمصريين، ان احنا عندنا ويل استابليشد ريسيرش سنترز اوريدي، ده موجود في مصر. والموضوع يمكن اختلف عن زمان، بقى في بيبرز كتيره بتتنشر من مصر based مثلا في الكورونا سيفيليانس موجود عندنا تيمز شغاله في مصر على الكورونا سيفيليانس. مينلي اللي انا شايفه ان الباحث محتاج التولز اللي ينفذ بيها البحث بتاعه، واحنا كشركه اناليسز all over Egypt احنا بنسيرف all over Egypt سواء مع الجامعات او طبعا جامعه مصر من الجامعات العريقه في مصر اللي شرفنا اننا بنتعامل معاها من زمن. والفتره الجايه في انيشيتيف من شركه اناليسز فور نوليدج ترانسفير، وبرضه البريباريشن بتاع الجونيور ريسيرشرز اللي جايين في الطريق ان هو انترنشيبس معينه على تكنولوجيز معينه افيلبل. ده اللي احنا نقدر نقدمه حاليا في اناليسز، وهنقدمه الفتره الجايه ان شاء الله ان سبورت للكوميونتي بتاع الريسيرشرز. اتشرفت بحضراتكم، انا متاسف بس على حوار البرزنتيشن. شكرا.
Thank you for this presentation.
Thank you.
And pleased to see you.
Nice to see you. Is there any question?
I don't know. I think we have not time for questions, but
okay.
All the afternoon will be dedicated to this technology and some other technology around DNA analysis.
Yeah. It's not just sequencing. يعني عندنا طبعا يعني work flows تانية أو techniques تانية زي المايكرو أري، زي الريل تايم، زي الكونفنشنال بي سي ار. كل تكنيك من دول بيسيرف الريسيرشر في حتة معينة. كل تكنيك من دول ليه الابلكيشنز بتاعته سواء ميديكال أو environmental أو researching studies. it's a wide profile of genetic testing لو احنا بنتكلم على الجيناتيك تيستنج عامه، بس هو طبعا الوقت مش هيكفي اننا نتكلم على ده كله.
Okay.
Thank you.
Thank you very much. Well, this afternoon session will be dedicated to DNA analysis by different ways. And the first speaker will be Dr. Tashin Shoala. He's yeah. Hello, welcome. Dr. Tashin Shoala is a specialist of nanobiotechnology. He is working in the College of Biotechnology in this university.
Yes.
And you can see the title of this talk behind me.
Thank you so much, Dr. Didier. Good afternoon, everybody. It's nice to see you here today. I would like to thank the organization committee for giving me the chance to be here with you to introduce my lecture, which is entitled to the nanobiotechnological approaches to manage phytopathogens. I think, do you have a remote? The pointer? Okay. So my outlines will be like, I'll give you a brief description or discussion about the nanotechnology, how can you design nanomaterial and different application of nanotechnology. And in the end, I will give you a list of application, which is my recent publication in how to use nanotechnology in managing phytopathogens. So nanotechnology is just a very old definition, like Richard Feynman, who was the first physicist who defined the nanotechnology in a meeting in the American Society of Physicists. And what he said is there is plenty of rooms at the bottoms. So he didn't define nanotechnology, but what he said, he mentioned nanotechnology without saying the main definition. But that was in 1959. And later on, Professor Norio from Japan, he started to define the nanotechnology by saying that the right definition of nanotechnology, the size between 1 to 100 nanometer. Just it's about designing and characterization of nanomaterial in the size between 1 to 100 nanometer. So nowadays, application of nanotechnology is not just using nanotechnology. We have to use innovative methods to apply nanotechnology. It is not about how to use it, how to prepare it, but now how can we innovate in nanotechnology? And this is what I'm trying to say, I will try to say today. So this, it's it's hard to imagine just how small it is. It's 1 billionth, just defining or making the material from a bigger size to smaller size. And as soon as you do this, you will change the physical character. As soon as you change the physical character, that means the material will have different function. Nanoscience and nanotechnology are the study and the application of extremely small things, and then characterize it using electron microscope to define what, if you approach it the normal size, and later on you will find the application of it. Nano is just a Greek word, dwarf, something small, and then technology visualize, characterize, produce, and manipulate matter of a material between 1 to 100 nanometer. There's different types of nanomaterial, like 1 dimension, very thin coating, 2 dimension nanowires, 3 dimensions nanoparticles, such as a very fine powder preparation. And with the very thin coating layer, you will be interested, you'll be surprised when you see a fruit in the market covered with a very thin layer of nanomaterial to protect it and preserve it for a longer time. As I mentioned, Richard Feynman was the first person who defined the nanomaterial. And then application of nanoparticles is just very fast and then going very quickly. And the way of using nanoparticles are going bigger and bigger nowadays in all the different fields. So the advantage is convenient, saving time. So within half an hour, you can prepare or sensitize the nanomaterial. Effective, but there is some disadvantages. I cannot be with my work on my side. So I like nanotechnology, but I have to see the disadvantages of nanomaterial. Environment problem. Sometimes when you use a specific nanomaterial, you might cause big problem in the environment. Killing creatures, so it's not specific. But with my work, I discovered a specific material which is biodegradable, naturally biodegradable, and safe to use in the nano size. And then polluting water, I completely agree with this, but there's some other nanomaterial like chitosan is not polluting water. We use it to purify water. How can we prepare the nanomaterial? There's 3 only main things, like using the physical method, chemical method, and mechanical method, a biological method. With the physical method, we use ultrasonication, we use irradiation, microwave, electrochemical, and then I have to warn all the researcher or scientist or the people, be careful when you use the microwave, because as soon as you heat the material, you don't know if you convert it to the normal smaller size. So the cell will lose selectivity, and then you might damage your body with using the microwave, because one of the very simple material or method converting the normal material from the normal size to the nano size using the microwave. So the microwave is really dangerous. It's not that simple. And then with other chemical methods like chemical reduction method, solidulation method, inert condensation method, and then later on the last one, biological method, is very simply, instead of using a specific material to reduce or oxidize a specific material to convert it to the nano size, we use extracts from plants or specific materials that could be extracted from plants. So here was the preparation of nanomaterials. Just we go from the bulk to the molecules, or the atom molecules to the normal nanoparticles, by using different methods like sputtering, chemical etching, thermal laser ablation, and mechanical explosion process. With the other way, like atomic molecular condensation, vapor deposition, solidulation method, and so on. There is different material that you can use to convert the material, the normal material from its normal size to the nano size. This is the description. So there's 4 main factors for the nanomaterials you have to consider while you prepare the nanomaterial. First of all, the size. We are talking a bit about very wide range of size of nanomaterial between 1 to 100 nanometer. So it's very, very wide range. But you will be surprised when you talk about every single size has every single different effect. So it's not that easy. We could say it's a range, but every single size could cause a different damage or a different effect, or could cure a disease. It just vary from one material to another. So the size is important. Then the nanomaterial, you have to know if it is negative, positive charge. So at least you will be aware about how to use it after that. Then you will talk about the shape. So different way of preparing the nanomaterial could give you a different shape, and all the different shape could give you a different results. Like for example, there is a specific researcher who could use, for example, silver nanoparticles. Use the chemical method to produce silver nanoparticles. Might be useful to treat for specific treatment, and might be harmful if you use it for a different or another treatment. And then the last one, the material itself. Are you using protein drug conjunction, then dimer, carbon nanotube, polymer, liposome, and so on, different materials? Those 4 factors are important when you design the nanomaterial. There is a very specific technique that I have to mention that they're using the nanobarcode. So that's a research trial has been done in USA. They are trying to detect many biological material just in one go. Like you could detect citrus virus, Ebola virus, different disease just in one go. What's the content of this technique? They use the tag of any type organic or non-organic material, and typically use gold, silver, any metallic nanoparticles, strips, and the cobalt already could be used. And then can easily be read with an electronic scanning device. So I will never rely in your eye or in your measurements. So I will rely in a scanning device to see if the right way to detect or to see if it's positive or negative. So the human error is just only avoided in this technique. Made through nanoscale electroporating of the materials into desired pattern. For example, here, this technique used the metallized template. As soon as you make the metallized template, it's sort of like incorporated in a different number, and these numbers could be read by the electronic device and could give you a different numbers for all these randomized metabolites or templates. And then you could use it to detect tobacco mosaic virus, citrus virus, and other diseases that could be harmful to the human being or the plant. So basically, they use it for the tagging food packages. How can you use this? If you go to any market, you will see the food is packaged with like different package, and then you will see the barcode. And as soon as the tissue itself of the fruits could release gas, the temperature inside the package could be raised to higher temperature, and then the barcode will be vary according to the state of the tissue inside the package. So if it is soft, you will see the color of the barcode, just the different colors, will be converted to, let's say, yellow color. If it is decayed, it will be like red color. So without opening the package, you could tell if the tissue is healthy or not, either using the scanning device or using your visual eyes to define the different colors. And this technique, like temperature changes, pathogen leaks, biomolecular trace tracing, you will see here the membrane, nitrocellulose membrane, adding the target DNA, and then pre-hybridization and then the nanobarcode, the nanobarcode with the barcoding, and then release certain colors, and it could tell you exactly or directly what is the infection, even if it is plant pathogenic or human pathogenic. Electrospinning is a very simple technique that you could use to spread out, just produce the nanoparticles in fibers. And I use this one of my experiments, like in I do research with one of my colleagues in veterinary Cairo University, and then this work was how can you cure a hernia operation by using a specific nanomaterial. And in my school, I use natural nanomaterial, not the metallic nanoparticles, because of the nanotoxicity. So very simply, electrospinning device, typical electrospun fibers, and then under pressure, this is a sort of electricity in here to produce the material in the nano size. And then the nanophotonics, just we can talk about it later on. Just I'll give you a brief about the nanophotonics. Like for example, using the laser. The laser, when you use the laser, there is a CO2 fractionation laser that you use it to treat any problems with the skin within the depth of 1 millimeter. But if you would like, this when you use CO2 as a source in this laser. But if you try to use it for something else, is like the cryo, they use halogen to penetrate further inside the body. That's why we use this. This is a very simple thing about the nanophotonics. I am running different research about how to use the light to treat plants. I'll go straight forward because I know that this is the main important part of my presentation. How can you treat plants, like application of nanotechnology in plant pathology? This is one of my recent research that's been published in November 2021. It has been published in Horticulture, MDBI, just a Q1 journal, and it's about nanobiotechnological approach to enhance potato resistance against potato leaf roll virus using natural nanomaterial, glycyrrhizic acid ammonium salt, and salicylic acid nanoparticles. Glycyrrhizic acid ammonium salt is it's a product or material from licorice root. So it's a natural material, biodegradable, could be safe if you use. Like the main idea here is just to enhance potato resistance against potato leaf roll. And as you all know, if the plant has been infected with virus, so that means you have to take this plant and just you have to dump it away or treat it or fire it. So you have to deal with it. But in my case, after the infection, I used to start to treat the plant with specific nanomaterial. And then the recovery stage started to occur, and then the plant started to reach the last stage. Here, I started to use a different technology. Before I use any material, in the previous days, what we used to do is just only testing the material and then see if it's good or not. But I relied on one of my colleagues who is dealing with bioinformatics to do the docking for my material and tell me if this material could be good or not. And what he has done, he tells me the story that glycyrrhizic acid is more efficient than the salicylic acid before I start. But later on, I tried to give it a go and see what's the difference between them. So one of my colleagues has done the docking, and the docking told me that the glycyrrhizic acid ammonium salt just have many connection or bonds with the coat protein. So that means the glycyrrhizic acid ammonium salt could interrupt the reproduction process of the virus. So that means could be a very good treatment to manage the virus. The salicylic acid was a little bit less than the glycyrrhizic acid. And then I tried to see the shoots and the roots or the number of sorry. Okay. The average number of leaves to see if the material itself has a good impact or negative impact on the plants itself. So it tells me that glyceric acid ammonium salt with the yellow color, it didn't affect the number of leaves compared to the control. And the other one was salicylic acid. Nearly glyceric acid and salicylic acid were nearly the same impact on the plant which has been infected with potato leaf roll compared to the positive control. And then I don't know why this. So when I have done the gel electrophoresis, it tells me that the potato leaf roll, the plants which has been infected with potato leaf roll, after I extracted the RNA and do the reverse transcriptase and do the real-time PCR or just the normal PCR to detect the presence and absence of PLRV, it told me that the PLRV decreased from infected plants after treatment compared to the control. And then I've done the expression level using the color synthase and MAP kinase. Why I'm doing those two genes in comparison with the reference gene, which is actin. Just those two genes specifically, they rely or they are responding for the resistance of plants against different stresses, especially MAP kinase gene. With the MAP kinase gene, just is responsible for two main things: root length and leaf area, increasing leaf area and the root length. As soon as you induce the MAP kinase, that means you will get higher leaves numbers and higher number of branches, and so that means the material itself is not harmful to the plant. This is something, it just tells me the hypothetical paragraph, which is, I don't know, is something wrong with the presentation. I don't know what's the problem. Here was a hypothetical diagram. It just tells me the scenario that could happen when you apply the material and the response of the plants the response of the plants in response to different treatments using the glyceric acid ammonium salt and the salicylic acid. So conclusion for the first paper or the first application, it told me that glyceric acid ammonium salt and salicylic acid nanoparticles could be perfect solution to produce potato plant-free virus. Okay, and then certain nanomaterial could suppress the PLRV virus via the induction of plant defense system. So we could use the different strategies by using either treatment for the infected plants or inducing the specific defense system, starting from the reactive oxygen species, ended by using the MAP kinase or any other pathways that could be induced in response to any other stresses. The other paper, which is about it's been published in the early 2021, just last January 2021. It's a pre-harvest application of salicylic acid induces some resistant genes of sweet pepper against black mold disease. So it was the first paper that's been published using salicylic acid in the nano size. Okay, so so I will give you a brief about it. I will not just go further because it's just I'm or behind the time. This been published in the European Journal of Plant Pathology, and the story about it, we use the salicylic acid nanoparticles to treat this disease, and was really successfully treated the infected plants and induced different resistant genes in the plant to decrease or delimit the fungi. I don't have more time to talk about the other publications. Maybe later on, I could give another presentation. You are more welcome to come, and I would like to thank you for your attention. Thank you so much.
Thank you, Professor. You accelerate a lot. It was nice.
Thank you so much.
Thanks a lot. Now we'll give the floor to an electronician, which is quite rare. It is my colleague Brice Sorly from the University of Montpellier. He will show you how to create device, analytical device for biological diagnostics. Thank you.
It's okay? You hear me? It's okay? Thank you. Thank you for the invitation first, and okay, thank you. So thank you for the invitation, and it's a great pleasure, a great honor to be here today. And I introduce myself. I'm associate professor in electronic, so I don't know nothing in biology. Microbiology, not yet. Sorry. So in this talk, I would like to present you the interest of the RFID technology. RFID is new for traceability, and and in this presentation, I would like to show you that it's possible to add some sensing part. Technology. I would like also to show you that it's possible also to add this sensing part and to give you opportunity to use this technology with your biotechnological skills to obtain perhaps new innovative tools. So this is a summary of my talk. First, I will present my lab. Then I will show you what is RFID, and the third part, how it's possible to connect RFID sensor and biosensor. And the last part will be on the application. So I come from Montpellier University in electronic lab, Institute of Electronic of Montpellier, and in the sensor department, and in my team, RFID and flexible electronic. And we work on sensor and RFID sensor. Montpellier University is 15 faculties and school, 50,000 students, 76 laboratories, less than 200 PhD students, and more than 1,500 researchers. So in my team, the objective of my team is to collect and transmit reliable and robust biosensor information through touchless technology for traceability, for diagnostic tools, for aid to decision, large volumes, and low cost. In three applications: food security, environment, and health. Our expertise is focused on materials manufacturing, structuration, substrate, and biofunctionalization, microtechnology and characterization on flexible substrate like paper or PET or PP, extraction of electrical properties up to 1 gigahertz, electronic simulation, and also wireless communication like RFID. We are also support few startups and help to technology transfer. And you have here two success stories, our team, Tageos company and Bontag company. So what is RFID? First, it is this is a comparison between barcode and RFID. you know what is a barcode. So you have here 1D or 2D data matrix barcode, and you have also a reader to scan this barcode. And the pros and cons of this technology: pro, low-cost technology, fast response, and cons, no sensor information and no global scan. In comparison, so with RFID. So what is RFID? RFID is radio frequency identification. So it's a wireless communication technology. How does it work? So it's called a new RFID, new barcode. RFID is new barcode. So you have an antenna. There is two components. Antenna, this is the materials like copper, conductive ink, or aluminum. And the most important part is an electronic chip. For example, you have a lot of manufacturer of this electronic chip as NXP, and also a substrate. And you have in this picture RFID inlays and RFID label. So if you see the picture on your right, you have the size of a chip. It's very, very, very small. So, what is the pros and cons of this technology? Pro, fast response. It's possible to add sensor. You can do a global scan, and the cons is not cheaper than barcode technology. So you can, with one request of your RFID reader, you can scan all the box in a palette, for example. So more particularly, you have different RFID technology. You have low frequency, and the principal application is for animal tracking. This is a passive technology. The speed is low for the communication. The cost is high, and the read range to read this is less than half meter. You have also the high frequency, HF. It's also a passive technology. Passive, you need to a request from a reader. Speed is medium. The cost is low. The read range is around less 1 meter. And the main application is for book tracking, for example, and access card. You have also the UHF technology, RFID. It could be, it's possible to have this technology in active. So active technology is this technology communicate itself, and also a passive. It's a passive technology. The speed is fast, the cost is lowest, and the read range is more than 6 meters. And the main application is for logistic. So the last frequency range, it's SHF technology, RFID. It's also active and passive technology, very fast speed, high cost, and the read range is more than 10 meters. It's also for motorway. Sorry. In our case, we will use this UHF technology in passive form. How does it work? So you have a wave emission of electromagnetic wave. This electromagnetic wave is from a reader, RFID reader, and the tag, so the antenna plus the electronic chip, capture the power and supply the microchip, and the microchip emitted wave back to the reader with and you can read the UPC global code a traceability number, a unique traceability number. So now, how is possible to add some sensing part? We have two possibilities. First, you have directly in the chip, you have a sensor, normally a temperature sensor. It's more easier. It's easier to produce this kind of sensors. And you have also external sensor. This technology is easy to obtain a sensing value, specific chip. So the cost is relative height. Monitoring is possible. If you do monitoring, you need battery. This is a reusable system, and only sensor could be disposable. And on the other side, you have cheapest technology. So in this kind of the sensor, the sensing part is added to the antenna. So it's not easy to collect information. Electrical impedance variation of the antenna according to sensing value. You use a standard chip, so the cost is low. This is a disposable system, and this is a totally passive technology, so the remote power supply. Application for this kind of sensor. Diagnosis health in food security. In diagnosis in health, you can notice 3 applications. We work on these 3 applications. E-dressing, so smart bandage, connected knee prosthesis, and tools for surveillance, prevention, and detection. And on the other side, with the low-cost technology, we work on the packaging, on smart packaging to monitor food spoilage. So now we want to see the few applications of this technology. First application with the sensor add to the chip. In this kind of sensor, in this application, this is a collaboration between startup Bontag company. This is designing of an orthopedic implant, prosthesis identification, and early unsealing detection. This is the objective of our sensor. And the specification was passive in vivo device with a 20 years lifetime, transcutaneous wireless communication by RFID technology, so without battery, and embedded force sensor for unsealing detection. So you have the picture of the prosthesis, and you have the implant location, and on the right, on the top, you have a schematization of our sensor. This technology is a technology readiness level around 8. And there is 4 patents on this technology and 1 startup created. And we work with them since 2014. The second example. So it's a RFID sensor, an APTA sensor, or nanobody sensor, or peptidic sensor. You will today, Dr. Mouez El Sadani present you an application of APTA sensor. So you have the target example: bacteria, virus, mycotoxin, MMP, metalloprotease. So you have a bioreceptor like peptide, aptamer, nanobodies, and you have a transducer to translate biological information in electrical information. So this is possible with this kind of tool. It's a capacitance sensor. So we have a functionalization on this surface, and we do an electrical impedance metric measurement. So you have electrical modelization of your biosensor, and these results we have. Sorry? Okay. Thank you. Thank you. And so you have some results here of this kind of RFID sensors, APTA sensor, nanobody sensor, and petit sensor. So on the graph on the right, you have correlation between the concentration of ochratoxin and electrical signal. So if you want more information, you have publication on bottom. So actually we work on H2020. The name of the project is COMBA, and we work on trypanosomes disease with this kind of APTA sensor. So the second possibilities for food security is so you have lowest RFID tag prototype with a biopolymer. The biopolymer is a layer of white gluten, and there is a correlation between variation gases and dielectric properties, with a variation of permittivity and loss. And with this kind of sensor, it's possible to us to follow the spoilage of, for example, a piece of meat. So the sensor is on the lid of your package. You have inside a piece of meat. There is a modified atmosphere packaging with 70% of oxygen and 30% of carbon dioxide. And you have on the right the reader and an application phone. So here, this is a correlation between food spoilage with the color of the meat, but also with bacterial count and RFID signal. And you can see at 5 days, the temperature is around 10 degrees here. And you can see that there is a correlation between RFID signal and food spoilage. So to conclude, passive RFID technology is for traceability and sensing, fixed and mobile reader, low range, passive technology, no battery, but also semi-passive with battery for data logging, for example. Electrical consumption is few 10 microwatt for communication, but also with passive technology, this is a remote power supply. There is a sensor functionality, small footprint, paper, plastic, textile label also. The cost is normally less than €1 for high volume. And there is a lot of application in health and in food safety. So, thank you for your attention.
Thank you, Brice, for this presentation. When I visited Gil's lab in Montpellier, I saw some props printed on papers. They are so small. Okay. They can print on the paper, on the big paper, some the props. Okay, the next speaker will be Mohamed El Hadidi. Okay, Mohamed, you are here. He's professor of biomedical science in the Wells City of Science and Technology. And I am very interesting, but you will tell us.
Thank you.
Because it is a part of my field.
I just want to double check these guys are working fine. Okay, so first of all, I want to introduce myself. My name is Mohammed Al-Hadidi. I'm a professor of biomedical science at the Will City for Science and Technology. My background is veterinary microbiology, so I'm interested in microbiology and microbial genomics, etc. So today I'm actually going to talk about one story, about one model that we actually use, how you use microbial genomics in order to track different zoonotic pathogens. So just like a simple question, how many of you know about microbiology, or have like some kind of background about microbiology? Just raise your hands. Good, perfect. So I'm going to tell a story, okay? I'm going to tell one story about one bug, which is Campylobacter, yet we use a lot of models in this context, okay? So do you know that 75% of the pathogen that we actually encounter are actually zoonotic, or actually the source are animals? Now you know, because you know about the coronavirus, right? You know about the SARS-CoV-2, so now you know how it actually originates from this pangolin that made us like for 2 years now with these masks, right? But 2 years back, if I told you like 75% of the pathogens that we actually encounter are zoonotic, you won't believe me. More than 200 pathogens are actually zoonotic. So what we learned also from the coronavirus is a risk somewhere is a risk everywhere. So the risk actually encountered in the China, and yet it's everywhere. You know, the miss is everywhere. So that actually encountered also in different bugs, you know, and in antimicrobial resistance. I recall, like, you know, then one of the most resistant genes, the New Delhi metallobeta-lactamase, it starts in India, and now it's everywhere, okay? So what I'm saying that a health threat anywhere can encounter disease don't stop at borders, you know? I'm not sure how to point this. Nice. Okay. I might need a help. Okay. You know what? I have my pointer already. Okay. So as I said, like a disease somewhere is a disease everywhere. So that actually encounters a One Health approach. And One Health is like, you know, we are always interconnected, like veterinarians, people working in environmental science, physicians, should work in an interdisciplinary collaboration in order to face any microbiological hazard. Okay, what is expected by 2050? Any person can simply be killed from diarrhea, a simple diarrhea, because there is no reserve in antimicrobial resistance. That's why I actually am interested in mechanisms of antimicrobial resistance. So we are actually investing too much effort on cancer research, yet by 2050, we can actually anyone can die simply by diarrhea that can be actually treated. And that actually Alexander Fleming actually expected when he actually discovered penicillin in 1941. So I'm going to talk today about one of my favorite bugs, which is Campylobacter. As I said, like I'm working in different gram-positive and gram-negative, but Campylobacter for me is really interesting that since I joined the Will City in 2018, I'm working in this bug. So it's almost a complete story. That's why I'm gonna just present today. So it's called a new food superbug. Why it's called like a new superbug? Because until 1975, no one discovered this Campylobacter, and since 1975, it's now the most common foodborne pathogens, more than Salmonella, Shigella, E. coli compound. So actually, combined with all the intestinal bugs, Campylobacter is a top-notch bug causing intestinal illness and diarrhea, and actually can cause some neurological symptoms. So I'm going to talk today about the story of Campylobacter, our research in the Will City on this freaking bug. Okay. هو مفيش pointer بس. برضو اوكي هو بس مجهد شويه. طيب so I'm going to talk about Campylobacter, about the background of Campylobacter. Campylobacter is a zoonotic pathogen. You can simply, when you eat poultry or eat chicken, you can mainly get Campylobacter if this poultry is not well cooked. Did you eat chicken today at lunch? You are at risk, okay? Yet, it's known that Campylobacter is the most common foodborne pathogens, yet we discovered, like, you know, since 1975 that there are other reservoirs of disease, like unpasteurized milk, cattle as a big reservoir. So I want to check also the difference between the reservoir and the host. So the reservoir is an animal that can carry the disease, yet والله point فين؟ أنا عايز point على السلايد. Okay. همشي بالاثنين. طيب, as I said, like the reservoir that you can carry the disease, but they don't develop any symptoms. Okay, back to the host or the human, they can develop severe illness and they can carry the disease and severe illness. So actually the reservoir of the disease, you can like eating, like as I said, like unpasteurized milk, eating poultry meat, etc. You can encounter the disease, and then there is no person-to-person infection with this bug. Yet with the excreta, which can contaminate the environment, you can encounter the disease. As a microbiologist, I was really interested in the epidemiology. So actually, like my PhD is veterinary microbiology and epidemiology. So I was really interested in the epidemiology of this disease, how I can use microbial genomics in order to better understand the disease transmission dynamics. Okay. This bug, what we know that is highly adaptive. So back like in the reservoir, it can highly adapt to the new environmental niche. It can require mutation, horizontal gene transfer. It can acquire genetic elements from the gene pool. We know about this. So it's one of the highly adaptive bugs, especially that it doesn't have a tight restriction modification system. So any DNA it can accept from the gene pool, it can accept and recombine with the genomic content. So that's why actually we know that this Campylobacter can adapt to the different environmental niche and which all these environmental niche can act as a reservoir for infection. In 2000, the first published genome of Campylobacter was published, and we know a lot of Campylobacter. This is by the second generation sequencing. Talking about the second generation sequencing, that was the first published genome, and it reveals like there is no repetitive DNA sequence. That's why we can do something like the variable tandem repeats. It's not like the gold standard here in tracking the epidemiology of this bug. Most of the bacteria actually are organized into an operon system. So different genes which are actually controlled by the same promoter. Yet in Campylobacter, the operon system is really rare. That's also what we expected by sequencing of this bug, that there is a lot of regulatory system that actually allow this bug to survive in different environmental niches. Therefore, the gold standard for this to track the genetic diversity of this bug is called multi-locus sequence typing. Okay, so the multi-locus sequence typing back like 5 years ago was the gold standard in tracking the epidemiology of Campylobacter. Campylobacter genome is like 1.6 base pair. Is 1.6 million base pair. When we do the multi-locus sequence typing, we usually use 7 housekeeping genes. 7 housekeeping genes which are actually not regulated for adaptation. It's like for normal metabolic functions, we use 7 housekeeping genes, we sequence these genes, and we have allelic diversity. And based on allelic diversity, we have now almost 300 or 400 sequence types and the clonal complex. So that was actually the gold standard, I would say. Maybe 5 years ago, they would say, like, if you want to track the genetic diversity of Campylobacter, use the MLST. It's an excellent platform in order to determine the genetic diversity among different Campylobacter populations from different niche. Yet, multi-locus sequence typing is actually using 3,300 base pair in total, which is like 0.2% of the whole genome. So it's unfair to use 1.2% of the whole genome to track the diversity. So we actually need something more can actually represent the high resolution power of the epidemiology. We use this technique. Like we use this technique back, as I said, like back 5 years ago or so. We use this technique in order to determine the genetic diversity from human cases and the most important source, which is poultry isolates. And as I mentioned here in this figure, the red is the human cases, and the green is the poultry isolates. And you can see some figure with these nodes represent the multi-locus sequence typing or the sequence type. So the one with the red and green means that this clonal complex is of infection risk. This column actually represents the lipooligosaccharide. So it does have like some kind of a weird cell wall structure with lipooligosaccharide of 5 types, A, B, C, D, and A, B, C are the most important one because it actually have a molecular mimicry with the nerve gangliosides. So it can cause autoimmune disease and can cause Miller-Fisher syndrome. So as I mentioned, like, 5 years ago, I was really interested in multi-locus sequence typing. I was a big fan of it. I was doing the multi-locus sequence typing daily. We used also like the second generation sequencing in order to determine some handful of genes which are actually published in literature. Like, we know like what are the genes in the Campylobacter which is responsible for resistance A, B, and C. So we characterized these genes and we correlated these genes and actually found some kind of with the second generation sequencing. I'm not talking here about the next generation. I'm going to talk later. With the second generation sequencing, we found like some kind of weird operon system, which is called the CME ABC efflux pump. Efflux pump is like the rotating door. So whenever antibiotics is going, it just get it out. So we found some kind of handful of genes, but not in the highest resolution that we could actually expect. As I mentioned, this was actually published also maybe like 3 or 4 years ago. Okay. Also, we applied this on Campylobacter coli as well, and we found correlation between antimicrobial resistance and different clonal complexes and some antimicrobial resistant genes. So 3 years ago, when I started my research platform at Zool City, I thought, okay, let's put this back and start from moving from the gene level, where I'm doing just like sequencing or multi-locus sequence typing or sequencing of some genes and correlating with the epidemiological context of the bacteria. Let's move to the genomic level. And this is the message I just want to deliver today. So moving from the gene level to the genome level. Does it provide a higher resolution into the epidemiology and the transmission dynamic of the bug? The answer is yes, because it answered a lot of questions that the gene level can't answer, like the microbial ecology, the antimicrobial resistance in Campylobacter, the core genome and the panc genome. If these definitions are not clear, the core genome are the genes that are present in 95 to 100% of the bacteria. And the pan genome are actually the core genome plus the accessory genome, which is plus or minus, present in a bacteria and not present in the other. The source attribution analysis, genetic elements with different hosts. So we answered all these questions over the past 3 years using the genome level. You are the expert in next generation sequencing. I'm not like the bioinformatician guy. So if you are attending this session for the bioinformatics, I'm not the Muhammad Al-Hadidi who is a bioinformatician. I'm Muhammad Al-Hadidi, the microbiologist. Okay, so I'm giving a biological background about like the next generation sequencing that you do the library prep, you assemble. You can assemble into contigs. We use some analysis that actually needs schemers analysis, and then we use these contigs or chemers for different analytical platforms, like the phylogenetic analysis from different sources or comparison between the local bugs with the global ones, define the core and accessory genome, the population structure, and we compare this with different bugs from different geographical origins. We do GWAS analysis if we actually augment our bugs with all the bugs on the different databases. So we do all this, and I'm going to show you some of our papers that was published actually maybe last year that implement the next generation sequencing in order to answer different questions about the epidemiology of this Campylobacter. So in this, I think this was published also last year in 2021, that actually we actually expose the Campylobacter into different environmental stressors. So we actually force this bacteria into different environmental stressors that actually act like a barrier between the reservoir and the human host, like the aerotolerance, refrigeration, heating, chemical decontamination, osmotic stress. We know, like food microbiologists know, that all these bugs are actually encountered in the food chain. So like it's what we call from farm to fork. Like from the origin until you eat the bug, there are some stressors that actually bacteria can encounter. So the question here is the bug really successful because it actually can resist these environmental stressors? And the answer that we found is yes. This actually, these clonal complexes that we know, the generalist, that can actually, is a successful pathogen, can actually encounter this stress, and it actually can resist the stressors that encountered in the food chain. We actually also screened the biofilm assay, and while biofilm assay, it's a mechanism of antimicrobial resistance, so the bacteria can coat itself with a layer of extracellular matrix where antibiotics can't actually penetrate this extracellular matrix, and we complement this with our data in next generation sequencing. it's a complicated curve, but it's pretty simple, okay? We define bacteria into 3 different categories. The HAT, which is meaning hyper-aerotolerant, it's really tolerant to aerobic stress because this bacteria actually is microaerophilic. It actually needs like small amount of oxygen in order to survive, like 5%. The aerotolerant, which actually can survive oxygen until 12 hours, and the aerosensitive, which are actually oxygen is really toxic for this bacteria. Surprisingly, we found like 65% of our bacteria is aerotolerant, which is meaning like oxygen is no more toxic for this bacteria. And this is the kind of evolution that we expect. This aerotolerant, we found very interesting result. These aerotolerant bacteria are cross-resistant. So once it's resisting oxygen, it can resist different stressors as well. So it can resist freezing and thawing, it can resist refrigeration stress, and these aerotolerant bacteria are actually high biofilm producer, which means that the bacteria can have a stress defense mechanism in the environment in order to be a successful pathogen. And like you can just type this title and you can find, and we can chat about, because actually the time is really limited. As I mentioned, we defined also the bacteria into generalist and specialist. Generalist means it's a successful pathogen, can occupy the reservoir and can transmit from the reservoir into the human host. Specialist, which are actually can encounter only one reservoir and probably it's not zoonotic. We found that the generalists, which are high infection risks, are more resistant to oily tolerance and can form a biofilm formation. So our hypothesis or our conclusion from this paper is, okay, this bacteria is successful because there are some clones which are named the generalists, can actually encounter different stressors and can resist to different stressors. And actually these stressors can actually act like a selective pressure in order to filter what are the infection genotypes to the human host. And these are the phylogenetic analysis of the generalists and specialists in the paper. Okay, another study that was actually also published last year by my graduate student, Nihal. Okay, we said like the multi-locus sequence typing is the gold standard. So the question here is, can we use the whole genome sequencing or the microbial genomics or the comparative genomics in order to design another gold standards better than the multi-locus sequence typing? So we use different typing methods that we actually use. The core genome multi-locus sequence typing, which is an upgrade of the multi-locus. The multi-locus, as I mentioned, you use 7 housekeeping genes. Here we use thousands of genes that you can use on typing. Can you compare like 7 in terms of the resolution and discrimination power with more than 1,000 or more? For this, so we use core genome multi-locus sequence typing, host segregating markers that we actually, it's like 15 markers. If you if you don't have the next generation platform and you want to do a PCR, it's unlogical to use thousands of genes. So you can use 15 genes, which are the host segregation marker, which is HS. If you don't have the, like in Zoid, we do have the next generation sequencing and we can do the library preparation and the sequencing. But I mean, like in like small lab, they can do this. So you can use the host segregation marker that we actually use for 15 genes. Most of them are accessory genome or soft core genome or core genome. We compare this with the CGF40, which are like PCR typing kind of. We found like 40 genes from the whole genome sequencing data that you can use as a barcode, presence or absence. With the multi-locus sequence typing, which is the gold standard before, as I mentioned, or the ribosomal multi-locus sequence typing. So all these typing methods are actually we implemented from our whole genome sequencing data and compared with the gold standard. Surprisingly, we found that the core genome multi-locus sequence data or multi-locus sequence typing is more discriminative as determined by Simpson index of diversity, and also it has a high concordance with the different typing methods. So we proposed in this paper that the core genome multi-locus sequence typing and host segregation markers might be more discriminatory. And hey guys, come and use this kind of genotypic techniques and forget about the multi-locus sequence typing that was used over 30 years ago. The nice thing that we actually used is the self-attribution analysis. So we have like a program called the structure that can actually determine, can we use this typing method in order to determine the source of infection? Remember the figure that I was showing, like, there are different sources of infection. Chicken is the main infection source, yet there are different infection sources. The pets, the ruminants are infection sources. So can we use microbial genomics in order to predict, hey, this is a source of infection? So we implemented this.
Mohamed, you have to finish now.
Sure. And we found that the beige is actually the ruminants reservoir, the blue is the poultry reservoir. So this actually indicates that in Egypt, poultry, and not only poultry, the main infection source, but actually the ruminant reservoir also is as equal in contribution with this. We use this also to predict some new antimicrobial resistant genes, and we found something really interesting, that the same lineage are showing the same homogeneous antimicrobial resistant pattern, which means that the survival for resistance to antimicrobial is not only because of acquiring different genes, but because also of successful lineage survival. بسرعه كده انا هشرح we compare this our bugs with the Oxford data sets. Why we use the Oxford data sets? It's like a long story, but it's too short to mention. But we compared the core genome, and we found that the core genome is as equal clustering with the Oxford data sets. And as I mentioned, the core genome are the presence in 95% of the bugs. We also compared the pan-genome analysis, and as you can see by these dots are the accessory genome between different isolates, which means that this bug is an open genome, so it can actually acquire different genes, which is meaning by the open genome. دي النقطة which I want to really mention for 1 minute. We actually measured the accessory genome clustering, and we compared the accessory genome clustering between Egypt and the Oxford datasets. And we found that the Egyptian datasets are actually acquiring more accessory genome, which means that the selective pressure from the using of antimicrobials in Egypt acting like a selective pressure for acquiring more accessory genome or horizontal gene transfer. This is also, we found that the multiple drug resistance for Egypt as compared to Oxford datasets phenotypically is higher. Finally, last but not least, and this was our last paper, we determined the toxin-antitoxin systems. And the toxin-antitoxin is another mechanism of antimicrobial resistance, where the bacteria, the toxin is actually, as you know, is stable, but the antitoxin is not stable. So the bacteria can actually resist the antimicrobial by actually secreting these toxins when needed, okay? But if not needed, it can actually neutralize with the antitoxin. So we identified 10 different classes of toxin-antitoxin systems among Campylobacter, and that was actually kind of novel because the toxin-antitoxin was usually used for some models like E. coli, etc. Finally, I would like to thank the key players, Nihal, who is my PhD student, Minna Sarafi, Dr. Minna Sarafi, Assistant Professor at Jewel City, our collaborators at University of Bath, UK, Sam Shepard and Ben Pescu, William Miller at USD Agricultural Research Institute, and my mentors in Belgium, Mark Hendricks and Kathleen Dirk, and my collaborator in University of Leon, Spain, Avelino Alvarez Ordoniz. So thanks to them for their hard work with my team. I would say that 95% of my work was actually attributed to this wonderful team. I'm just like this boring PI who is writing grants and just checking who is doing what. But actually, all this work is because of this wonderful team. Thanks to my team, to my co-founders, and thank you so much for this.
Thank you, Doctor. I have three information for you. There are 2 conferences that will be not given by Professor Al-Sayed, who didn't come, and Professor Alexander Azea, who cannot give his presentation. It means that we will go outside earlier. And one more information, the coffee break will be removed, skip. That's the bad news of the day, but it's like this. And now we'll prepare the presentation of Dr. Mansour Akan, who will give his presentation online. Could you prepare the online presentation? Now you have the photo of Professor Mansour Khan. Professor Mansour Khan is the Director of Research and Development of a company, if I understand well, EMDA International in Saudi Arabia. And he will give a talk on NGS, the next generation sequencer, who is a machine, who is a revolutionary system in the analysis of DNA, actually. Professor? Yes, we have the transparency. Professor, you are ready?
Yeah, good evening, ladies and gentlemen. Thank you for inviting me, and I'm honored by Missouri University of Science and Technology. So let me introduce myself. I'm Dr. Mansoor, working as a director in India International Establishment in Saudi Arabia, as well as regional sales manager in Moleculon, New Zealand. So let's start our talk with the question, what is sequence? So the answer for this question is that to find out the sequence of bases A, T, C, G of a particular gene or a region of DNA is DNA sequencing, while in RNA is RNA sequencing, and in amino acids it will be protein sequencing. So sequencing in the last past decade, nucleic acid sequencing has increased a lot as the technique is widely available and accessible to research and clinical lab all over the world. The most common method for single gene sequencing is Sanger sequencing, which is also known as first generation sequencing. The method involves random incorporation of di-deoxynucleotides, which are chain terminating when encountered by DNA polymerase. So this slide shows you the picture of di-deoxynucleotide stop synthesis. So move to the next slide, which is showing the Sanger sequencing map. In first step, it contained a mixture of like PCR with di-deoxynucleotides, and in the second step, it shows the separation by capillary gel electrophoresis due to fragmentation. The smaller the fragment, the faster will be the elution. And in this third step, the laser excitation and detection by the machine, the sequencer. So we will move to the next slide, the importance of DNA sequencing. So the discovery of DNA sequencing in past decades brought many breakthroughs in various science domains such as gene expression studies that leads to protein structure and function, finding out mutation to detect diseases and disorders. Personalized medicines then is useful for the doctors who prescribe medicine, and then we reach to forensic for dispute cases. As we know, every technique have pros and cons and limitations. So obviously the Sanger has. The Sanger, the limitation Sanger have is that it can read only 300 to 1000 base pairs per sequence. And the human genome is about 3 billion bases. So it will be very costly if we do Sanger to do human genome. So it is a very expensive technique in terms of human genome. The second thing, which is also not very good, is often not very good in the region of first 15 to 40 bases because of the primer binding. So the third issue is, as I already described, it's very expensive, and then it's time consuming as well. If you go for 3 billion bases, then it will be ages. So we will know after these limitations, then the next generation sequencing, or we can call the second generation sequencing, has been introduced. So with the completion of Human Genome Project, the demand of robust and fast technology for sequencing increased, which led to development of NGS. This technology uses massive parallel sequencing, during which millions of fragments of a DNA from a single sample as sequenced and in less time. This picture shows you that what is the difference or like between Sanger and NGS. In Sanger, you catch one by one, one gene one time, while in NGS, you can catch a bunch. So move to the next slide. These are some platforms where which world scientists or clinical lab or research labs are using. Next to in the next slide, we'll talk about next generation sequencing again. The development of NGS platforms allows sequencing technology accessibility to more labs, which in turn increase the quality and quantity of research and clinical diagnostic. The two main widely used platforms are IronTorrent from IronTorrent PGM and Illumina MySec. So that the methodology of these platforms includes template preparation, sequencing and imaging, and data analysis. So here is the some common steps for NGS methodology. The first common step for all of those platforms or all of these platforms which we've seen in the last previous slide, sample extraction, DNA fragmentation, and adapter ligation are the common step for all the platforms. While if you go in Roche and ABI, amplification is done by emulsion PCR, while in Illumina, the amplification is by bridge PCR. So in this slide, it's NGS platform. There is a comparison of the platforms. Like if you see by read length, Roche has 700 base pair read length with the accuracy of 99.9%. With other have little less, but that is also fine. This is not a big difference as in terms of accuracy. The read length or read per run in Illumina, if you see in Illumina, it's 3 billion, while the other Roche is 1 million and IronTorrent is 5 million. They are less. So if you go in this area and it's 3 billion, the time is also a major factor, and it is 1 to 10 days to do this 3 billion. So this has got a more famous position in the market for having this kind of facility in Illumina. While, as I described before, that every technique has pros and cons. So there are some advantages. The disadvantage of this technique is that it is very expensive. So it's hard to afford it right currently. It might be cheaper in the future. So we move to the next slide. The application of NGS in human, if you see in human heredity and complex diseases, it can be used in cancer research, it can be used for population genetics, it can be used, and if you go in drug and R&D, it can be used for biomarker discovery and validation, disease and animal models. If you go in plant and animal research, it can be used for evolution and diversity, for microbial. The industry can use this technique it's like agriculture industry or pharmaceutical and other as well. So we move to the next slide. These are the future application of NGS. In this future application, you can say that these will be in the coming years. This will be established. These techniques will be established in coming years. So we'll move to the next slide. This slide is very important for us as well as for might be you, that it explains that what we are and why we are here. This slides give you an idea that the company which is providing these facilities in through us is BGI. BGI has got 4 domains, covering all 4 domains, that genomics, transcriptomics, epigenomics, and proteomics. So in these all 4 domains, they are providing another 4 like covering all 4 sectors, which is NNGS, NGS, MS, and microarray. In regard this, BGI have its own platform as well, which is BGI SEC 500 and BGI SEC 50. So if we go to the next slide, these domains are integrated, and these like as I showed you before, that they are interconnected as well. That if you go in DNA, gene sequence, gene variation, gene regulation, while in RNA level, it gives gene regulation, gene expression, while in protein, it's protein expression, and then it leads to network. So we move to the facility here in BGI. In BGI is a huge facility available for the scientists, which have got, you can say it's 1,000 sequencer they have, 5 Illuminas they have, 2 MySec they have, and like as I said, 5 Illumina, it's 5 HiSec, and many more. So if we move to the next slide, this is what we say to the people that BGI or IMDA International in Middle East is providing one stop and gives you total solution. Okay, that means like that if you provide, like we provide you, if you provide us the sample, we provide you sequencing service, we provide you consultation platform, we provide you to understand the scientist's problems, and we provide the solution corresponding to various budget levels as well. Like we can do the budgeting, the cost effectiveness of these techniques. So we provide you, you provide us the sample, and we provide you everything under these circumstances. So next, move to the next slide. This is the workflow chart for WGS, quality control, which is whole genome sequencing. This chart explains that at every step we do quality control, like from sample providing, like sample preparation, it will go for quality control. Then library construction, we go for quality control. Then sequencing, we go for quality control. Then data, quality control. It is linked with quality control. Then delivery, it will be with quality control. So if we move to the next slides, this is how it works, like whole genome sequence workflow. That you provide a sample, like we do sample preparation. We do library construction, we do sequencing, we do raw data output, and which leads to bioinformatics. And if the sample, what you provide is the sample type. You can say there are many samples type you can provide us, like genomic DNA or saliva, blood, FFPE, and there are many more. So we can move on that as well. So the next slide is give you, these are the bioinformatics tools which can be used, or which we are currently using for your bioinformatics analysis. So the next slide, it's a very interesting slide. It shows that if you go on a dinner and you like something in that dinner, then we say sequence it. If you go in a shopping or in somewhere where you like something useful, you find something useful, we say sequence it. Okay, then if you go in a zoo and you like some animal there, you can sequence it. If you go in a garden, you like some plant or flower, you can sequence it. So if we move to the last slide, this slide is very important for us and for you as well, that it explains that the material which are used before NGS or before Sanger. So these are very basic or a very important material and tools which scientists are using all over the world. So we are supplying these materials, like the materials of, you can say, the bench top equipment, extraction kits, purification kits, PCR enzymes, real-time reagents for real-time PCR, plus electrophoresis reagents. These all we are supplying through our supplier in New Zealand, which is Molecule On, and they also deal in the plasticware, the PCR strips, PCR tubes, or filter tapes. They are our main supplier for anything which is before sequencing or before next generation sequencing. So we, as we said, and we are still repeating it again and again, that we are the service provider, or we are providing you the total solution under one umbrella of IMDA International, which is based in Riyadh, Kingdom of Saudi Arabia. And this is all about this till now, and I think it's the time is running off, and it's enough from my side. Thank you very much for your patience, and we'll see you sometime. Thank you very much. Bye bye.
Hello. Just a remark, this presentation was recorded. He's not online with us, actually. I will not speak with Kim because it's a record. Okay. Thank you so much, Mr. Khan. Now we will shift to another speaker, who is Mrs. Noah Radwan. I don't know. Are you here? Yes. Okay. Noah Radwan is Associate Professor of Clinical Pathology
Hi.
at the College of Medicine at
Hi.
Cairo University.
Hi. Thank you. Thank you. First, I would like to thank the organization committee for inviting me in this conference. This lecture will be about could Sanger sequencing fill the gap in the prognosis of COVID-19 outcome? Sorry. On 7th of January 2020, researchers isolated and sequenced in China from patients with severe pneumonitis a novel coronavirus, then called SARS-CoV-2, which rapidly spread worldwide, becoming a global health emergency. This figure shows the stages of the infection with COVID-19. The early stage, there will be an early infection with viral replication, asymptomatic or mild to moderate symptoms. The second stage is the pulmonary stage, where the viral replication and affecting the lung, causing pneumonia, dyspnea, and the CT will show glass opacities. The third stage is the hyperinflammatory phase, in which there is a host inflammatory response leads to pneumonia, sepsis, respiratory failure, acute respiratory syndrome, and cytokine storm. The common symptoms in COVID-19 pneumonia are fever, dry cough, shortness of breath, myalgia, fatigue, lymphopenia. The uncommon symptoms are headache, hemoptysis, diarrhea, confusion, nausea, and vomiting. The most common complications to COVID-19 pneumonia are acute respiratory distress syndrome, secondary infection, ventilation-associated pneumonia, septic shock, acute renal failure, hypoxemia, and acute cardiac injury. The virus can induce an excessive and apparent host immune response, and this is the key in the COVID-19 infection, is the host immune response, characterized by an upregulation of pro-inflammatory cytokines resembling the clinical and serological features of cytokine release syndrome. Cytokine release syndrome is a life-threatening emergency associated with high mortality, thus as early identification is crucial. COVID-19 invades the lungs and causes inflammatory signals to be released from dendritic cells, monocytes, macrophages, and causes cytokine storm, which leads to ARDS, acute respiratory distress syndrome, multi-organ failure, hyperinflammation syndrome, and the worst of all, death, of course. The cytokine storm observed in inflammatory stage of COVID-19 has been reported in other autoinflammatory diseases, such as the macrophage activating syndrome and familial Mediterranean fever. Familial Mediterranean fever, which is known by FMF, and COVID-19 show a remarkable overlap of clinical symptoms and similar laboratory findings. Both are characterized by fever, abdominal or chest pain, elevation of C-reactive protein, and leukocytosis. In this paper, which is published in September 2020, there was a question raised about the similarity between familial Mediterranean fever and COVID-19, which are whether are friends or foes. In addition, colchicine and interleukin-1 inhibitors treatments that are effective in controlling inflammation in familial Mediterranean fever patients have recently been proposed for off-label use in COVID-19 patients. MEFV is the gene responsible for familial Mediterranean fever. It's located in the short arm chromosome 16. And familial Mediterranean fever is caused by different mutations in this gene, which encodes a protein named pyrene. Pyrene gene is expressed mostly in neutrophils and in eosinophils, monocytes, dendritic cells, and fibroblasts. The main function of pyrene is involvement in inflammatory response and in resolving inflammation by autophagy of regulators of innate immunity, thus controlling the inflammation, which is the key in controlling the inflammation not to cause a cytokine storm. There are more than 300 mutation variants located on the MEFV gene. Most of the mutations are in exon 10 between amino acid 680 and 761. So the gene responsible for FMF, which familial Mediterranean fever, which is MEFV gene exon 10, was investigated in 39 patients attending Qasr Al Aini Hospital due to COVID-19 pneumonia, with no previous personal or family history of FMF or familial Mediterranean fever. These patients were sorted according to the triage protocol in the Ministry of Health of Egypt. There was 19 patients in moderate cases in which the oxygen saturation was above 92% and was with some risk factors like diabetes, hypertension, and etc. 20 of patients was admitted to the ICU for severe cases in which the saturation of oxygen was below 92%, the respiratory rate was above 30, and lung infiltrations was above 50%, and admitted to the ICU. Surprisingly, 90% of patients who had variants in exon 10 of MEFV gene, either heterozygous or homozygous, had only moderate symptoms, while most of patients with severe symptoms has no variant at all. These are examples of the heterozygous variants that was found in the moderate group of patients. This one was the first one was substitution of alanine by serine in the position 744. And here is the silent mutation in the position was 706. And this the last one is substitution of valine with alanine in the position 726. This letter to the editor was published in 2020, raised the question about could MEFV mutation carriage state as a protective rule in the COVID-19 pandemic? In the paragraph, it was stated that, on the other hand, this inflammatory syndrome perhaps was a protective factor against frequent and lethal infectious diseases. And in another paragraph, it was stated that death rates are lower in countries where Jews, Arabs, Turks, and Armenians predominate. This data implies that lower incidence and milder disease course of COVID-19 in these countries may be due to increased prevalence of MEFV mutation carriage stated in these populations. elucidating the genetic determinants of COVID-19 severity and susceptibility to SARS-CoV-2 infection would allow for a proper stratification of individuals according to risk, so that those at higher risk would be a guide to personalized therapeutics. So sequencing of MEFV gene could give us a clue about the course of COVID-19 infection, especially in hospitalized patients, and helps us to determine the prognosis of these patients. Thank you. Okay, thank you.
Thank you so much for this
Thank you.
interesting information about COVID. Now we will shift to the last speaker. Who is Dr. Rehab Abdallah, Mrs. Where she is? Okay. Alors, Rehab Abdallah is an adjunct professor, and she in Egypt, but she's actually in Germany in postdoc at the Max Planck Institute.
Hello, everyone. I'm just checking the pointer. Thank you. Okay. Switching, yes. First of all, I'm really happy to be with you today, and thank you for the nice invitation. And I really hope that my talk will be light, and it will not be too much for you guys because of the long day and so on. So I am Rahim Abdullah, and today I'm going to talk to you about the different meta-omics techniques. Okay. And how can we use them to unravel or understand the unique microbial community that are present in Egypt terrestrial and marine habitat. Okay. Could you just hold a minute? It's not working, and laser, right? Yes. Laser مش هاي. Two slides, we won't need the pointer for them. Okay. طيب, let's start the talk by asking you, what would you think, and what is the first thing that would come to your mind when I say sea أو البحر؟ إيه أول حاجة هتيجي؟ غالبا حاجة شبه كده صح؟ A nice view, sea, water, and a really nice sand to relax. But not all people will think about this when anyone tells them the sea. Okay, so I am one of the few people that when you tell them the word sea, they will think about microbes like this, such as Plegibacter ubiquity or Chlorococcus, responsible for around producing around 20% of the oxygen that we breathe. So microbes in the sea and ocean make around 90% of the biological mass, and they are actually responsible for producing a lot of metabolites that are responsible, for example, for the smell of the sea. The smell of the sea is actually a product of microbes. Okay, so this is not the only importance of marine microbes. They are actually, as I said, make around almost half of the oxygen that we breathe. They are also having a huge role in the global carbon, nitrogen, sulfur cycle, and they are have a huge role as sink, or they are metabolizing carbon dioxide and methane, which, as you all know, are having a greenhouse effect, or producing a greenhouse effect. They are greenhouse gases. Yes, thank you, shukran. Okay, And most importantly, I think for the majority of the audience, that they are producing, or they are producing a lot of bioactive compound and enzymes that could be useful in industry, in research, and specifically in the pharma industry if you're interested in antibacterial and anti-cancerous agents. So as a microbiologist, the first thing that you will do if you're interested in an environment such as the oceans and sea, you'll probably go to conventional microbial technique. You're going to take a sample from the sea that you're interested to, a sediment sample or a surface water sample, and you're just going to culture it on a plate. Now, since 2006, we started to move a little bit away from the conventional microbial techniques, and we started to study the entire environment. If you are a microbiologist, you know that the media will not grow all the microbes that are in an environment. And that's why it's very important that we use a technique such as metagenomics or metatranscriptomics to help us understand the entire community and each and every member in the community, and also look for gene of interest that might be present here. And these members that are harboring this gene cannot grow on the plate. Again, it's well known that around 99 or 89% of the microbes cannot be grown on a general culture or culture media that we use in the lab. So I will talk about 3 approaches that we use for metagenomics. The first one of them is one that majority of the scientists in microbial ecology and environmental microbiology use, is the targeted gene approach. An example of this is 16S ribosomal RNA gene sequencing. However, other genes could be targeted as well. The first, how to do this kind of sequencing approach, it's very simple. You would just go into the environment that you're interested to study, collect the sample, extract the DNA, and then do a PCR amplification on the regions that you want to study. For example, in this case, it will be the 16S ribosomal RNA. You use the NGS sequencing technique that were described in the previous lecture, and then you analyze the data. This technique is very nice, but the problem with it is that it has limited information. If you're interested in a gene of importance to you, or a gene responsible for the production of an enzyme, for example, this is not going to help you. So this is mainly for classification of microbes in an environment. Now, if you're interested to study the genes responsible for the production of enzymes and bioactive compounds, and understanding also the metabolic potential of the microbial community you're interested in, then you need to go to shotgun metagenomics. And here, instead of doing a PCR amplification, we fragment the DNA and we just put it into the sequencer, and we sequence everything that is there. Then we do an assembly-based approach, and then we look at the assembled region or the contigs, and we look for enzymes or open reading frame of our interest, depending on your own interest. Some people like me would even go one step further and try to bind those contigs together to get something called metagenomic assembled genome, and then we look at the metabolic pathways, and we do metabolic reconstruction on these genome. So we try to understand each single microbe, what they are doing, and if they are even communicating together with each other. Now, this is on the genomic level. There is a third approach that is very nice for people who actually don't like to work with sequencing a lot, and they don't have that bioinformatics background, strong bioinformatic background, which is functional metagenomic approach. Now, this approach is based on, in a state of, after fragmenting the DNA that you got from the environment, in a state of sequencing this, you're just going to put it in an expression vector. Is it phosmid or back or plasmid? The majority of the functional metagenomic library are made on phosmid, but other people use back and plasmids as well. After you add the fragment of the DNA or the random fragment of DNA into the vectors, then you will transfer it into E. coli cells, and then you grow these E. coli cells and do functional screening. So if you're interested in lipases, for example, then you use tributyrene, for example, and then if the E. coli contain a lipase, it will clear the tributyrene on the plate. and then you will have your own enzyme of interest. This approach is very nice because it helps us look at genes in the environment that if we try to culture the bacteria doing them, we might not be successful. So we're taking the gene and we're putting it inside E. coli and seeing if E. coli could express it. Other gene of interest could also be tested, such as tolerance to pH or heavy metals and so on. Okay, so this is basically the 3 arms for the DNA-based approaches. There's also an RNA-based approach, and this approach is basically used when you want to look at an activity. If you have a community in the environment, and this community you want to change, for example, you want to look at the change in environmental condition and how this change affects the microbial community. Then if you use DNA, it's not going to be very meaningful or significant. It's always better to look at the mRNA. There's also the protein-based approach and the metabolite-based approach. Here you're looking at the product of the microbial community, and here you're just going one step further from the metatranscriptomics. Both proteomics and metatranscriptomics could be used to decipher the activity or the response of the microbial community to an environmental change. Now, the project that I'm going to discuss will be mainly focused on the DNA-based approach and a little bit on the RNA-based approaches, because those what I have been using during my PhD and after the PhD as well. So yeah. Okay, so the question now, how can we use these techniques to understand and explore different marine and terrestrial microbial communities or microbes? So the first project that I would like to talk with you about is the Red Sea Marine Metagenomic Project. This is a project that started actually in 2008, but we still have people working on the data generated from that project until now. The project focused on 3 different brine pools along the brine pools in the Red Sea. Brine pools are like depression in the sea floor, and they usually have a very interesting characteristic. They usually have high salt concentration, high pressure, high temperature, abundance of heavy metal. And because of this, they represent a very interesting environment to explore unique microbial community that we wouldn't see, for example, in normal agricultural soil or in fresh water, and also their product, because we expect that the product of the microbes living in such a place would have unique characteristics such as resistance to heavy metal and ability to degrade, for example, substrate under high temperature. So the first thing that we worked on was a generation of a functional metagenomic library. My colleague Yasmin worked on identifying and isolating a heavy metal resistant thermophilic esterase. 2 minutes? Okay. Esterase from the Red Sea brine pool. Yeah, this thermophilic esterase was thermophilic esterase it had its maximum activity at 65 degrees Celsius. It was resistant to heavy metal, tolerant as well, so it resists salinity or sodium chloride, and also have tolerance to detergents. This enzyme have a lot of application in food processing, paper industry, so pulp industry, and detergent industry. Now, what we have done is that we recharacterized this enzyme, and we looked further into that enzyme, and we were able to see that this enzyme is actually cyclostable, and it's not only resistant to heavy metal, it actually get activated. So the activity increased in the presence of certain heavy metals such as cadmium and copper and so on. And this paper is actually under submission or was just submitted. The other project was by, again, a colleague of mine, where she looked at different microbial community along the coast of the Red Sea. And she have a lot of important points, but I just want to focus on the conclusion that she found that there is unique hydrocarbon degrading bacteria, around 75% of the total bacteria present in two hypersaline lakes, Solar Lake West and Saline Lake RM, are actually unique, and they are responsible in the degradation of hydrocarbon. And based on her work, we started a new project. This is a very big project where we looked at the microbial community in those two hypersaline lakes, and our question was, how does the halophilic microbial community degrade hydrocarbon contamination under high salinity? Because these Solar Lake and Saline Lake, their salinity could reach around 18%. This was a shotgun metagenomic approach. This is actually was a sample from a summer sampling trip that was made in 2021. We just received them. We have 12 samples, but each sample is replicated. So we have replica in these 12 samples, and we have a humongous data, around 120 gigabytes. So around 60 million read per sample that we are analyzing to understand this question and to decipher the microbial community and the different enzymes involved in hydrocarbon contamination. I wanted to talk about metatranscriptomic a bit and my work at the MPE, but due to time limitation, I'm just going to skip this. And yeah, in conclusion, metagenomics is again used to identify who is there, basically what kind of microorganism is there. Also help us to understand the metabolic potential of this community and provide a resource for novel enzyme and bioactive compound. Metatranscriptomic and metaproteomics, on the other hand, could help us to identify the change in the microbial community that occurred due to a change in environmental condition. And this could actually have an application in agricultural soil, especially rice paddy soil, and for example, how they could respond to soil salinization. And based on this and the response of the microbial community, we could see how this could affect the soil health and fertility. And at the end, I just would like to thank my group at AUC. I mean, this is only the subset of colleagues that have helped me during the project that I have mentioned, but our group is very bigger. It's humongous. It's way bigger than this. I would like to give a special thanks to Dr. Rania Siom, as well to my group at the Max Planck Institute in Marburg. I would like to thank both of them for their contribution to the papers, and thank you.
Thank you so much.
Thank you.
We could say that we finish in time. That's not the truth. We have seen in this session what is wonderful now in microbiology. We can make a lot of catalogs of microorganisms. When I was young, 40 years ago, when I studied microbiology, we had already a lot of microorganisms, many, and nobody knows exactly how many microorganisms we have. And now with the sequencer, we have a little bit more. And the life of the microbiologist is becoming difficult and difficult every day. The advantage that we have seen some young researchers that speak this afternoon, that show us also that we can study the metabolism of microorganisms with these techniques, which is new, really new. The future of this technology is in the study of the metabolism of microorganisms. Thank you for all.
Would like to thank you all for being with us today. I know it was a long day, but actually it was filled with science. So we are looking to see you tomorrow, inshallah, in a fresh state. But before leaving you, I would like to apologize for any inconvenience having this day, because organizing such event is like we are looking for the perfection, but the perfection is the ultimate goal. But on the route to perfection, you can find some obstacles. But we apologize for any inconvenience having today and look forward to see you tomorrow, inshallah. Thank you very much.